All right, I have started sharing my screen once again. I hope it is visible to everyone.
Yes, Srigori, it's visible.
Thank you. All right, we'll be starting with the presentation in just 5 minutes. And meanwhile, we are waiting. It would be great if the participants who have not introduced themselves yet to introduce in the chat so that we know about background and what are your research interests. And thank you so much, Daniel, for interacting with the participants and
yeah, you're welcome. No problem. You're welcome. Hello, Shugari.
Yes, Daniel, we'll be starting in 4 more minutes with my presentation. We are waiting for the rest of the participants to join.
Okay. No problem.
All right, so we have with us Mohammed Abdul Rob, and he's a student of biochemistry and molecular biology. Thank you so much, Abdul, for introducing yourself. Can you also let us know about your research interest? And what about the rest of you? I'm curious to know about you as well. I can see that several participants have just joined with us. It would be great if you can introduce yourself as well in the chat.
Sorry, are you referring to me?
Sorry, who is this?
My name is Abdul Rahim Akintunde.
Yes, yes. Could you please also let us know about your research interest?
My research interest is in the area of environmental health issues. What are the latest trends, or what are the latest issues in the environmental health area?
And how did you get interested in the field of bioinformatics and data science? I'm curious to know too.
Well, thank you for that question. There is a friend of mine who is into bioinformatics, and we've discussed extensively so much, and I see a nexus between this area and my area of research. And being a researcher, which is a multidisciplinary researcher, I tend to explore new areas like the bioinformatics so that I can see where I can contribute to knowledge, especially in the area of the media popularization and awareness creation. So that's my major area. That's my major interest in this bioinformatics.
All right, thank you so much. That was pretty good to know about. And can you also let me know, how would you rate your bioinformatics and data science skills? Are you a beginner, or are you already familiar with the field?
Well, I can just say I am a beginner because starting, or probably I have a few or very few background in it. So let me just say I'm a beginner, so that by the time I now upgrade, start getting exposed to this area, then I can now pick or develop myself in that direction.
All right, thank you so much, Abdul, for introducing. So I'm curious to know about the rest of you as well. Please feel free to introduce yourself in the chat with your educational background and research interest, just like Abdul did. So with that, let us start with today's symposium. Let me first turn on my camera. All right. I hope I'm visible. Okay, so hello everyone, a very warm greeting to one and all. It is my great pleasure to welcome you all to Omics Logic. So to begin with, we must ask why biological data analysis. Data analysis is an essential component in any biopharma or healthcare industry. Data analysis in biology has already moved out of the domain of conventional statistics, and it is expected that a student of biology is acquainted with basic concepts of modern data analysis tools. Biological data analysis also has practical applications in healthcare, agriculture, and mitigating climate change. And as this field progresses, biological data analysis poses several challenges for both students trying to learn it and faculty trying to teach it. So how do we at Omics Logic tackle these issues? We'll get to that in a short while. So before we begin with today's symposium, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The Omics Logic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leveraging our online training resources, adapting them to the needs of students and researchers around the world. This training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. And in this presentation, I will explain the current challenges and limitations in biological data analysis, describe how we aim to address these challenges with the Omics Logic Learn portal, describe Bloom's taxonomy for learning progression, and how it can be used in biological data analysis coursework. Explore the Omics Logic portal and how it utilizes Bloom's taxonomy in structuring its coursework, pipelines, projects, and finally describe several outcomes of the Omics Logic portal. So what are some of the challenges that the field of biological data analysis is currently facing? So to begin with, this field is constantly growing and developing new technology and software to improve processing time and power. The data sets that are analyzed are usually very large and take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. And in a university setting, students have the opportunity to learn the background information of designing and implementing a biological data analysis project, but they do not receive practical hands-on experience with the full workflow due to the limitations mentioned previously. Similarly, faculty faces the challenges with being able to cover a diverse range of topics if they are specialized in a certain area of biological data analysis, or if they are unfamiliar with data analysis in general. For example, a professor whose research focuses on oncogene expression might not know about the metagenomics. So to overcome these challenges, the Omics Logic portal was developed. This aims to standardize, organize, and systematize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework. That is, materials for courses are usually developed in-house, taught sporadically, and are rarely updated. The Omics Logic portal introduces new material that keeps up with new technology. And these courses also need to be organized because the challenge is to combine several different topics together. The flow and transition between the subjects also need to be developed. This is addressed at Omics Logic through introductory material being woven throughout the coursework, the last challenge we face is systematization. So how can you know that someone is stuck and needs help, or allow someone to run forward? So to address this, Omics Logic uses standardized modules and interactive sessions, beginning with mastering tech terminology and concepts to implementing these concepts with real data sets in a project-based approach. So here is an overview of the learning progress that takes place at Omics Logic. In order to create a clear path of skill development, Omics Logic employs the Bloom's taxonomy levels of learning. So for those of you who are not familiar with this approach starts with the basics, that is, students recall facts and terminology, and they're able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The students will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, that is the ability to design and create original work. So how do Omics Logic resources help advance these above mentioned points? Omics Logic is a portal for practical and theoretical learning of biological data analysis. It is composed of courses designed to take you through the theoretical knowledge. Omics Logic takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with biological data analysis is, Omics Logic is going to be a very useful tool for gaining a baseline knowledge of the theory behind biological data analysis, analysis of large data sets, and introduction to basic coding languages like R and Python programming, or even beginning a data analysis project. So here are some of the courses that we have on the portal. These 7 courses were taken from the core Omics Logic curriculum available online. So beginning with an introduction to the application of biological data analysis and an overview of data types, the student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, data science, and cheminformatic concepts are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the students to gain the conceptual knowledge in order to begin their own projects. Omics Logic expands upon these basic curriculum with specialization track projects. Biological data analysis methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various biological data analysis methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available data sets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insight from complex omics data. The Omics Logic portal also gives students an option for analyzing a given data set. So there are code-dependent paths or there are code-independent paths. These data sets and associated publications can be used in several ways. For instance, you can learn biological data analysis without any coding at all. And for that, in each project, we explain the analysis pipelines on the user-friendly Bioinfo platform for big data analysis, which I'll be introducing in my next slide. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python programming in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs, where experts share their expertise and support research project that is developed by you. So here is an example of the code-independent pipeline. The pipeline builder interface on the Bioinfo platform is a color-coded pipeline that establishes a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. So first of all, the data is put in as an input file, and then analysis can be chosen based off the type of research that you want to carry out. And each analysis is separated into 3 colors. You have blue for pre-processing, red for mapping, and green for quantification. And each time you click a particular algorithm, a pop-up will show up that contains additional information that is useful for building the pipeline. The ease of the color-coded buttons allows the program to be run without any coding glitch, and interactive output allow easy visualization of the data. And several demo pipelines with real data sets are available for students to practice the workflow, including demos on genomic variant calling of cancer cell lines, 15S amplicon analysis of diet and its effect on the microbiome, and even a pipeline examining phylogenetic analysis of Ebola virus. So after using the Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their knowledge, remember the understand levels of Bloom's, and then apply that in a project. Here comes the apply, analyze, and evaluate levels of the Bloom's taxonomy that I was talking about in my previous slide. Then check their comprehension with levels and badges, certificates of completion, and tests. Similarly, faculties should be able to assign the Omics Logic portal to their students with the understanding that it will give them a holistic and comprehensive knowledge of bioinformatics, beginning with Biotech molecules and on to designing their own question-based project. The Omics Logic also employs a hybrid approach. I'd like to request the participants to mute yourselves so that, thank you. Omics Logic employs a hybrid approach combining learning and research to ensure that students are building off of the first levels of Bloom's taxonomy. Omics Logic track progresses with activities like pipeline, coding, and literature reviews, quizzes on topics, key terms and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then students are able to continue their knowledge base with the research assessment, making sure that they are able to understand the methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should also be able to ask questions based on the data exploration that they carry out. This leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and finally find a solution for it. Biological data analysis combines life science terms with data science skills, which allows researchers to answer life questions with reproducible data analysis methods. So now you might be wondering what comes after all the training and completion of the coursework. The answer is possibilities are endless. So to begin with, a majority of the students who enroll for the programs are focused on publishing their research work. So for instance, Prithvi Vaja, a biomedical science graduate student who worked on assessing the underlying reason for development of gray matter lesions in multiple sclerosis patients at the transcriptomic level, went on to publish her work. In Immunogenetics, which is an open access journal that focuses on the genetic research areas of autoimmune disorders such as multiple sclerosis, rheumatoid arthritis, and so on. And another interesting student publication that I want to share with you all is that of Supanta Dev, a genetic engineering student. He studied the effect of malaria vaccine on the transcriptional profile of patients to determine its efficacy and understand the molecular mechanisms underlying the protection this vaccine provides. He also went on to publish his work in Genomics and Informatics Journal, which is the official journal of Korea Genome Organization. Presently, just like Supanta and Prithvi, we have several other students who are working on their publication, like Nivedita Arambam, a biotechnology graduate student. She is working on transcriptomic study of cancer treatment on patients and its ever effects leading to relapse. Like her, we are looking forward to several other publications from our current students. Further, you also have the opportunity to participate in poster presentation competitions, such as part of the Omics Research Symposium that we are having right now, to win exciting cash prizes. So this symposium is an international research symposium that brings together experts from all around the world. The student projects are not just limited to graduates, doctorates, or postdoctorates. For instance, Andre Modano Sosa, a high school student at Mexico, presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potamkin Prize and the 4th award in the Biomedical and Health Sciences category. And this program can also be integrated as a part of existing university curriculum. For instance, Fine Biotech offered the Machine Learning for Biomedical Data Python course, which was integrated into the System Medicine's graduate program at Georgetown University. The lectures were prepared considering the interest of the program director to address the challenges that is relevant to clinical application, biomedical research, and of interest to the industry, including big pharma and biotech firms. The training ended with students submitting their research projects on the Omics Logic Learn portal. So I would like to request all of the participants to take a look at it. I'll be sharing the link in just a while, and you will come across several student projects on infectious disease, neuroscience, psychiatric disorder, oncology, machine learning, and so on. And these are very inspiring to look at that students have carried out these projects. And these projects can also be showcased for higher studies and international university applications, like some of our research fellows. For example, Dilara Taikin, she got accepted to the Erasmus IMBRC program in marine biology. Dhruv Behra got accepted to a precision medicine graduate program at University of Manchester. And Anusha TA just recently got accepted to a PhD program in metagenomics at a university in Sweden. So faculties and researchers have also collaborated through our training programs to publish their research work. And our latest collaboration has been with a team of researchers at Noguchi Memorial Institute of Medical Research at University of Ghana. And they carried out a computational-based study to understand variants associated with sulfadopterin resistance in Plasmodium falciparum. And it is also possible to pursue an entrepreneurship and business innovation track, just like a team of industry researchers from Biologic Support Services, a biotechnology company in Nigeria. Their aim is to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term by developing a test kit. The entrepreneurship track is not only pursued by researchers, but by students as well. For instance, a group of undergraduate and postgraduate students worked on developing an early detection kit for early detection of neurological disorders with a specific focus on Alzheimer's disease. So now, if you are interested to learn more about where these courses are and how you can view various student projects that have been carried out, I would like to direct you to Omics Logic Learn portal. It is free and easy and free for anyone to register, with many courses available at no cost. So to view the various courses, example projects, and student projects, you need to first sign up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, and a brief bio about yourself that will consist of your education background and research interests. And as you can see, under the activity tab, the mentors can view your progress. The courses you are currently completing will appear under the courses tab. Finally, after the completion of your coursework, the certificates will appear under the certificates. That was all about that I wanted to discuss in this presentation. With that, let me walk you through on how you can sign up on the Omics Logic Learn portal. I'm sharing the link in the chat box for the participants. I'm sharing the link in the chat box for the participants who have not signed up yet to sign up and explore the various projects and courses that I was just discussing in my video. And if you have already signed up on the portal, please put a yes in the chat box so that I know that you have already an existing account on the portal. All right, so for those participants who are joining with us for the very first time and want to get started by signing up on the portal, you can click on the create an account option that you see here. First, visit the learn.omicslogic.com/signup link, and then click on the create an account option. And once you click here, you will be asked to enter your name, email ID, and password. You can either sign up using any of this method, or you can also sign up using any of your social media accounts. So this could be your Google account, Facebook account, GitHub account, Apple account, or even Twitter. So I'll pause for a couple of seconds if the interested participants wish to sign up so that they can sign up. And all right, I do have a couple of yeses in the chat. If you're facing any kind of technical issues while signing up, please let me know. Our team is here and we'll be happy to look into it as well. Or if you want me to explain any of the steps that I was discussing about signing up, please let me know that as well, and I'll be happy to explain. So with that, I'll just pause for a couple of seconds for the participants to sign up, and then we'll resume with our navigation through the. All right, I can see few yes in the chat box. So I'm hoping that I can proceed ahead so that I can show you how you can. So since I already have an account on the portal, I'm just going to click on login now. I have entered my existing email ID and password, and I'm just going to click on sign in. So once you sign in, you are seeing the courses page. So here, when you scroll down below, you will see a search bar that says looking for a specific course. So here, if you are to enter your key area of research interest. So for example, we had a participant who was just mentioning that he's interested to learn about bioinformatics, but he seems to be a beginner. So for such beginners, I would like to direct you to introductory courses on bioinformatics, such as when I enter the term introduction, it will show me the very first course 1, Introduction to Bioinformatics. So this is a very free course, and I'd like to encourage all the participants who do not have a previous knowledge of bioinformatics to take it. So this course on biological data analysis is an introductory course that covers the topics of big data bioinformatics and its uses in basic research, such as healthcare and biotechnology and pharmaceutical industries. And the topics are arranged into 3 segments, as you can see here. The very first lesson focuses on big data bioinformatics, then bioinformatics in healthcare, and translation bioinformatics. So let me just open one of the lessons and I'll show you. Each of these coursework comprises of asynchronous content and video tutorials, quizzes, assignments, and even interactive visuals. So you can take a look at it at your own pace and navigate and take a look at all of the courses that are there. So another free introductory course that I want to direct you to is the course 2 Bytes and Molecules. So this happens to be one of my very favorite courses on the portal, since it introduces you from the very beginning by introducing you to the concepts such as citizen science, molecules, data and life, code of life, and then the course focus on particular topics such as structural biology, genomics, transcriptomics, metagenomics, and my favorite one happens to be the Space Omics one, which comprises of several interactive video tutorials from astronauts explaining their work and even linked to some of the publicly available data sets that the participants can take a look at. So that is one of the courses. And I'm also noticing that there are several participants among us who might be interested to learn about Python and R programming. So for such participants who want to get started in this field, I'd like to direct them to Python course 1, Getting Started with Bioinformatics. So this course is covers the introductory basic fundamentals of Python programming, from teaching you how to import packages such as NumPy and Pandas to reading DNA sequences using Python code, to calculate genome-based frequency, and even creating reverse complex. So whether you're from a biology or from a computer science background, this course will help you equip with the required bioinformatics skills to help you get started in Python coding. And at the end of the coursework, students will be able to create, write, and execute Python functions, and even create very beautiful data visualization using Python. So that was all about the Python coursework. And I'm also assuming that there might be participants who might be interested to learn about our program. So for such participants, we have the very first coursework, that is our course 1, getting started with bioinformatics. So this course is for those participants who want to learn how to load data and visualize it, as well as analyze genomic sequence and find patterns that can help us interpret the language of DNA, RNA, and protein. And the course also introduced basic concepts in R of data structures and on working with a unique type of data sequences. So that is all about that I wanted to show you about the courses. Apart from that, there are several intermediate coursework that I encourage participants who already have a prior background knowledge of bioinformatics to explore. So for instance, if you are someone who is interested to learn about genomics, then I would like to direct you to course 3, genomics. And if you are someone who's interested to learn about transcriptomics, then I would like to direct you to course 5, transcriptomics. Like this way, if you type any of the key areas of research interest that you're interested to learn more about, that will show up. So apart from the coursework, you can also see several example projects that have been taken from high impact research publications that break down the research work that has been carried out by other scientists into simpler terms, so that it is beginner friendly, and for those participants who want to get started in research will be able to understand how they are carried out. So that was all about navigating through the courses. You can browse through it at your own pace. And now I'd like to take you to the how to set up your account settings. So to be able to do that, once you sign up, you will see a blue bar that is welcome back with your email ID. When you click on this, it will directly take you to your account settings. So here, if you are to click on the button that is profile, you will be able to edit all of the features that I was just talking about in my presentation. So that includes your name. Make sure that your name has to be in proper capitalization, since the certificates will be printed out. And you can add a bio about yourself that will consist of your educational background and research interest. And you can add a social media link as well and upload a profile image. So once all of that is complete, it will be showing profile completion as 100%. So now you will also see the activity tab, like I mentioned, where the program mentor and the community manager, they will be tracking your live progress on the portal. The courses tab will keep a track of all the courses you are currently enrolled in, as well as show the coursework progress that you're making. And under the programs tab, you will see several ongoing programs that you must have enrolled in. And finally, once you complete the required coursework with associated assignments and quizzes, you'll be able to receive all of the certificates under the certificates tab. And finally, one interesting tab that I want to show you is to click on the projects tab, as you see here. So this is the place where we have all the collection of student projects that I was explaining about in my presentation. And these topics range from oncology, infectious diseases, precision medicine, neuroscience, data science. And some of these projects have also been published. So you can take a look at all of that and explore at your own pace. So that was all about that I wanted to introduce the participants to about the Omics Logic Learn platform and training. So with that, let me introduce you to the today's Omics Research Symposium. The Omics Research Symposium is a 2-day online event aimed at bringing together a network of experts, educators, and students from around the world to learn from each other, interact, and discuss advances in the field. Discussions will highlight challenges involved in research using computational tools for big data, opportunities in this outgrowing field, and applications of advances in various fields. So today we have the opportunity to hear from esteemed experts from all around the world about their groundbreaking research, and I'm certain that their talk will leave all of us with a great appreciation of the importance of biological data analysis and the need for further research in this area. So without further delay, please join me in welcoming our very first speaker, Dr. Mohit Mazumdar. Dr. Mazumdar has over 12 years of research and industry experience. He completed a master's in bioinformatics from Jamia Millia Islamia and PhD in computational biology. And he is an expert in machine learning in application to biomedical and chemical data. He's currently heading the global business development for Pine Biotech USA and contributing to biological data science research and education by collaborating with universities in India and Africa and mentoring participants from various programs by providing research support. And today he'll be introducing us to the Omics Logic Asia and how it is working to advance biological data science education in the region. And with that brief introduction, now I would like to pass on the stage to Dr. Mohit Mazumdar. So, over to you, sir. Sir, it looks like you're on mute.
Oh, I'm sorry. Thank you, Shrigari, for the introduction and telling me that I was on mute. And it's great to see you all here today with us. I'm really excited. Every year, we prepare and wait for this time for our Omics Research Symposium to be able to meet with you and learn from the experts in the field of biological data analysis for research and industry or business. So for many students, it's an opportunity to compete, showcase their research work, and get recognitions and prizes. So I hope you all are as excited as we are to hear about these cutting-edge approaches in life sciences, as well as challenges, not just from our team, but from experts who have spent their entire lives struggling with these type of challenges, and more importantly, coming up with great solutions. Also, it is great to see a much broader appreciation of data in life science domain, as well as a great excitement about the potential impact of biological data analysis, bioinformatics on areas touching the lives of everyone. So medicine, vaccine design, genomic surveillance, and even space travel and space omics. So at Omics Logic, our mission is to simplify bioinformatics and advance research through these different modules that Shrigari was just showing you, and the intuitive multi-omics research platform. We are working with students, researchers, and we are providing research support and also working on several research projects for biomedical drug discovery and well-being of and healthcare and well-being of human beings. So eventually, or ultimately, we are working for a future where we can contribute to enhance human health and well-being by enabling biological research and discovery with relevant data solutions support. That's our journey, and in that journey, I'm really happy to have an amazing team with us. And I wanted to use this opportunity to briefly introduce our team who will be there in the chat and who will be helping you with any of the technical challenges that you might be facing in the next 2 days. So since 2019, we have launched Omics Logic in India with initial collaborations at several universities where we piloted our training and worked with faculty to develop programs that addresses these major gaps in biomedical data science education. And since then, we have been working with students from partner universities going through the training and project work, and faculty who have showed interest in utilizing the collaborative research platforms and collaborative workforce to incorporate project-based biological data science and curriculums and coding curriculums into their into their coursework, into the students' training of undergraduate, postgraduate, pre-PhD, pre-trainings and all. So in India, it has been a thrilling journey, and this experience has enabled us to work on variety of challenges that comes with any educational training pertaining to translational research or pertaining to something that has been not so much well established. So when we started, we were facing a lot of challenges in terms of awareness of omics research, awareness of data and data science. So with that agenda to bring awareness about data-driven healthcare research, our team reached out to universities, and our agenda was to able to our offerings was to able to provide computational infrastructure and flipped curriculums that the faculty can use, and the resources to be able to get high quality data driven research accessible to everyone, like undergraduates, postgraduates, PhD students, which is one of the biggest challenges in bioinformatics, doing high quality bioinformatics. So this engagement has helped us collect data and tremendously improve our own learning models. So today I will be showing you some of those outcomes from what we have experienced in Asia, in our programs in Asia. So after many programs coordinated with universities and thousands of students from India, we saw that there is a gap in skills that helps students to effectively integrate into a research experience and thinking more critically about data and applying appropriate analysis methods to the right data. So to address those gaps, we came up with our educational collaborative model where every student we think needs to be enabled with data skills and analysis logic to succeed in their research, excel in their career, and be ready for the world of data that we live in. So schools, universities, colleges, how they are needed to be equipped to help faculty and student learn and use this data. And as I'm saying this, we understood that it's a massive responsibility on our hands that can only be accompanied by an ever-growing community. It's just something that we cannot do alone, and that's why we need all of you to be able to do work towards the similar goals that would be helpful for any nation and even the entire world. So our appreciation to all our academic partners who have come forward and joined hands to bring the awareness, the tools, and the technology and an international standard to the training and research in biomedical data science. So in this presentation, I'll explain our initiatives and highlight the partnerships and collaborations from Omics Logic, Bioinformatics and Data Science Training in Asia, and this upcoming expert sessions and discussions on day one of this Omics Research Symposium. And we will be discussing about the university training programs that we recently did. We will start from there. And this entire 1 hour is mostly dedicated to an introduction and to showcase how these different types of educational and research awareness is going to be very helpful in the future to develop capacity. So after learning from our experience and working with universities in India, we were able to start a new chapter in Bangladesh. And this was a new challenge that our team has taken up after the university reached out to us with their interest. And it was interesting to know that the Genomics Center in Jashore University, which is a very renowned university in Jashore, Bangladesh, is kind of now going into more detailed sequencing roles. So right now, they have used a lot of RT-PCR and generated a lot of data from there during the COVID pandemic and have the centers. And now they are going for sequencing capabilities, and they were interested to learn and bring that capacity to be able to utilize sequencing capabilities with the right kind of analysis and outcomes. So what we did is that we kind of wanted to incorporate also the learnings or the curriculums that they have developed at Jashore University, integrating with our own curriculums that we are going to offer, which includes statistical analysis methods and visualization, use of popular machine learning techniques, which Shri Gauri was showing you in the previous presentation. So students were able to learn about public data repositories and various file formats, bioinformatics for storing and efficiently processing omics data, and also the topics from the university that focuses on, the RT-PCR analysis, phylogenomics, and protein modeling. So this was an integrated program that we recently completed. And in this program, the great thing about such programs is that the collaborative effort of both the team members, both the teams and both organizations help students to really get the most out of this. So we had a collaborative team of faculties from Pine Biotech and Jashore University helping students go through this entire experience and make the most out of this. So over 100 students joined this program, and over 50 students completed it with very high points, and many of them, and then the rest of them were able to start and still are progressing through the Omics Logic community. Similarly, I mean, in India, as I already showed, that we have been working with several university partners, and we had this opportunity to work with a group of faculties. So Honorable Vice Chancellor, Professor Deepa Sharma at IIT University, Meerut, and Assistant Professor Dr. Vatsala Tomar help students with this industry academia partnership to work collaboratively and integrate modern and industry approaches in healthcare and biomedical research, and using this interdisciplinary skill set of different departments at the university and developing a research ecosystem. But all of this starts with the faculty. So we did recently a faculty development workshop, and in that faculty development workshop, which is to help faculty to come up with different types of research areas integrated into their own research. So our university training programs are recognized by some of the top research and educational institutions in the US. And we are glad to bring this high quality and effective experience to those who are interested to work collaboratively in Nigeria, in India, in other countries where we can have a collaborative framework to be able to do this and sustain all of this. So this workshop series focused on powerful scientific data generation techniques, and also some of these techniques which we were really going to deep dived in is next generation sequencing data analysis, and also the publicly available data that is helping a lot of, like at least thousands of researchers in discovery to translational research. So we covered the basic next generation sequencing process, where we discussed about the fragmenting DNA, RNA sequences into multiple pieces, to bringing to adding adapter and sequencing and libraries to be, and also which is more focused on different types of data that you would generate for cancer, for infectious diseases, for neuroscience, giving an idea of these risks, how big data is integrated into day-to-day research that one could be able to do with the capacity that we were able to provide. This year, we extended our community by starting to work with departments interested in applying big data analysis for pharmaceutical research and innovation. And that's where we collaborated with NIPER Hyderabad to assist the students of pharmacology and pharmacy to be able to integrate big data analysis like genomics and transcriptomics into their research, into their project work. So a group of students from BTech, from bachelor's and master's, joined the program and completed the training on genomic data analysis and transcriptomics data analysis under the guidance of mentors from Pine Biotech, as well as Dr. Vasundhara Bhandari, Assistant Professor, and Dr. Shashi Bala Singh, Director of National Institute of Pharmaceutical Education and Research in Hyderabad. So we had a lot of programs that we were able to do in some of these areas in Hyderabad, including Government City College. And going back to the relationships that we have already developed and continuing developing these programs for our partners who are open to integration of industry relevant training and development for their undergraduate and postgraduate life science education to provide job opportunities and internship opportunities. We went to Amity Jaipur. We met with their students and their faculty, and we were able to do some workshops for the students from biotechnology, bachelor's and master's and MTech in biotechnology, food technology, and microbial technology and applied sciences, who were eager to learn about genomics, metagenomics, and how different types of omics data could be used in food technology and agriculture. And we also had that opportunity again to work with the administration at Amity University, where the emphasis on was to do course structure. So how industry related courses can help students to directly integrate with the industry once they finish their degree. What would be the delivery plan? How the course will have, what kind of learning outcomes will the course will have, what would be the program learning outcomes, how this will be mapped with Amity and external contributors, and would facilitate in research and development. So we had roundtable meetings, we had panel discussions where different industry experts came and helped students understand that what they need, and also help the university understand that where the need is and what kind of technology and courses are required for students to be able to go and get those job opportunities which are really available in the market. So that's about Rajasthan. So in Rajasthan, we had an amazing opportunity that I wanted to show you all and talk to you all about, which was partnering with some top research and academic institutions in India to develop training and development programs for working professionals, as well as for students to upskill and integrate more modern data literacy and expertise. So we recently collaborated with IIT Jodhpur, Department of Biotechnology, Biosciences and Bioengineering, where we collaborated with Dr. Pankaj Yadav, Professor Mitali Mukherjee, and we met the students and we interacted with them. And we had a lot of discussions about different types of projects that IIT Jodhpur and these students are working on, collaborating on metagenomics research and other research which are relevant to harsh environments in the Thar Desert, and also working on new projects that would be about the capacity development in India in this area of next generation sequencing and its application. So this led to something really new, a new program that we have envisioned and we are going to start in this summer, which is a certificate and diploma, PG diploma program from IIT Jodhpur, in collaboration with Omics Logic, Biotech, and Premas Life Sciences, where there is where there will be an opportunity for students to not only learn the data science part or data generation or how to analyze data and interpretation, but to also do the wet lab experiments, do the data generation, and also learn from the experts in IIT Jodhpur and Pine Biotech in an intensive 6-month certification and 1-year diploma course that would which would enable students to get job opportunities and get integrated with some of the partners who will be there in this program. So it's something that we are really excited about, and we are hoping that we'll be telling you more about such opportunities. So that's about Rajasthan. And I wanted to take you from Rajasthan to little above Punjab. So we have a wonderful experience collaborating with universities in Punjab, and we have a team there working in Punjab, including our team members who are present here, Sparsh, Ms. Sonalika, Ojasvi, and Akshita, and we have been able to reach out to some of these universities, do online and on-site classroom studies and workshops. And the focus of such collaborations vary from university to university based on the need of the university. So these modules that we have been showing you, and the modules that we have been developing in collaboration with researchers are being utilized by university departments or university students or a PhD student based on their own research interest. So the focus with Chandigarh University CGC Land Run is to be able to provide basic bioinformatics training to final year students who are working on research projects. So our team at Chandigarh facilitated the ceremony there and helped with the administration to start the hybrid bioinformatics training and research. Similarly, we were really happy to start working with Punjab University, and we have been connected with Punjab University for the last 3-4 years, trying to work with Dr. Ashok and their other collaborators. And recently we collaborated and recently we are working with the Department of Microbiology, where our focus would be metagenomics analysis to be able to help the students who are going through microbiology honors. So that's one program, and another with the Department of Bioinformatics to be able to help them with next generation sequencing, data analysis and interpretation using machine learning and data science tools for model generation. So these are some of the important areas that Punjab University focuses on, and they have asked us to collaborate and help develop these cutting edge programs for their students. So another aspect for us to be able to do this is to be able to help students with opportunities which are beyond education and training, which is more relevant to job opportunities. So we keep on doing as industry academia meets with the university, giving opportunities to the students for job as well as internship, and connecting them with the network that we represent, being an international community, to be able to go for higher education in some of the top universities, or go for an industry experience internship or a job opportunity which they are eligible to. So our team has been meeting and going to these different events and helping students to develop such career in their after completing their degree. So and continuing this with other universities. So we also collaborated with Chandigarh University, and their focus was to help the biotechnology and computer science students who are doing undergraduation and post-graduation life science degrees, to give them an opportunity to work on BioML and also learn about data science application in R, and also to do biomedical data science in Python, because the computer science students, they are really fascinated with how the application of computer science is now such a big application of computer science is there in life sciences. So from application development to data wrangling to data transformation, all of that is something that really interests the computer science departments. And that's why this collaboration and understanding how to write and execute your codes and use Jupyter notebooks and start doing exploratory analysis and do different types of data sets using Google Colab, and check those codes and make sure that all of these components are related to some objective that is either translational or has some research nobility in it. So that's one of the reasons that we have been working with these different universities, that actually describes a pipeline. So that's like coming towards the end of my presentation. I would like to show you a case study. So this is with Amity University, Uttar Pradesh, and this is the pipeline that we have developed and designed. So it's a multi-departmental pipeline, which includes, it starts with the technology training, like the next generation sequencing, as you saw in Sri Gauri's presentation, and learn about different types of technologies like chem informatics, health informatics, NGS, and then data science. So this is technology training. And then it involves like different stages in technology training. So you learn about, gene expression, then you learn about differential gene expression, then you learn about machine learning, how all of these are different stages and steps. So there are advanced training steps. And then there is research specialization, where your interest is, maybe precision medicine, maybe oncology, maybe neuroscience, infectious diseases, agriculture, metabolic diseases, and AMR, whatever public repositories data that is available. There is some bioinformatics application or value that we will be able to extract. With that idea, we wanted to show that with the incorporation of faculty here, we will be able to work and use the student capacity to work on noble projects and showcase them in symposiums such as this. And in this entire process, we are able to provide the career pathways, and this entire pathway includes, going through the developing skill sets for people who are just interested in developing skill sets, for people who are interested in research and higher education and job opportunities. So there are different career pathways that that came out of this, like finding a lab for academic purpose, working with a team in an industry, in academic internship, and job opportunities which are relevant for these fields, and the companies that are working on these fields, and the academic labs that are researching in these fields, and as well as entrepreneurship opportunity like the hackathon that Sri Gauri was showing you. So all of this effort is through constant meetings and discussions with the administration, with the faculty, with the director, with the VC, with the assistant professors who have initiated these conversations initially and have helped us build this relationship and develop different types of projects. So we saw some patterns which I wanted to show you quickly, that students who are going through this initially had some ideas, but their ideas got more and more defined. They used a lot more new projects and topics. There were a lot of students from different types of backgrounds, and it is kind of becoming easier for students to now do this and go through this in a structured manner, which is otherwise very challenging considering how the world of bioinformatics is, and it's open source. Many of the things are free, but to navigate through it, you need experience, you need expertise. So it's a structured way to go through this navigation and get a starting point and then explore by yourself. So this is not something that makes you dependent, but it makes you aware, and it makes you ready for the future and to take up new challenges in research and in the industry. And we are really happy, and we are really glad to have been partnered with many of these international and national collaborators, and we are happy to be a part. Happy that if you are interested to be a part of this community, which is helping with data awareness and helping reaching out to more students, will definitely will be something that would be helping for capacity development, as well as the importance of healthcare in our lives. So including our industry partners, I'd like to thank all of them for coming on board and helping students with these opportunities that they are bringing, with the connections and contributions. So with that, I'd like to invite Sri Gauri again. Before that, I'll just tell you that we did this hackathon, and this is an example of how industry is contributing to all of this, working on these relevant topics that are important for the market, and then trying to come up with a minimum viable product using research. So we have other industry partners, and this is my presentation for tomorrow, where we have focused on industry partnership and how industry is helping in and contributing in healthcare and in bioinformatics. So with that, I'll just conclude my presentation, and I'd like to ask Sri Gauri to invite the next speaker, and let us know if there are any questions. I'll be happy to answer them in the chat.
Thank you so much, Dr. Mohit. Let me just quickly share my screen. All right, so for those participants who missed the session right now, let me just quickly summarize the topics that were discussed. So this session was designed to introduce the audience to various Omics Logic programs in Asia, and Dr. Mohit Mazumdar shared his insight on recent collaboration with Jodhpur University and IIMT, and even providing customized curriculum for NIPA, Amity Jaipur, IIT Jodhpur, and Punjab University, amongst all, and how these efforts are bridging the existing industry-academia gap in biological data science research and education. So thank you so much, Dr. Mohit. And if the participants, any of you are interested to participate in the partnership program, please feel free to reach out to us. I would like to request our symposium moderator to share the link to the program page. So with that, let me introduce all the participants to the next session by Dr. Andrew Lin. So as we all know, the ongoing COVID-19 pandemic has had a profound impact on public health worldwide. And the development of effective vaccines against SARS-CoV-2 has been a critical step in controlling the spread of the virus. So with that, building on the insights and knowledge that we gained from the very first session about ongoing biological data science research and education in Asia, I'm excited to introduce you to our next speaker, Dr. Andrew Lin. Dr. Andrew Lin is currently a professor at the School of Computational and Integrative Sciences at Jawaharlal Nehru University, where he has been a dean and coordinator of the Center of Excellence in Computation Biology and Bioinformatics, as well as Director of Communication and Information Services. He has served on the Board of Studies in Bioinformatics at Banaras Hindu University, Banistali Vidyapeeth, Central University of South Bihar, and Pondicherry University. His areas of research are predominantly in computational biology, biomedical data science, where he works on the development and deployment of computational methods applied to biological sequence, structure, and systems analysis. So without further delay, let us welcome our distinguished guest, Dr. Andrew Lin, as he takes the stage to discuss his work on modeling sequence evolution in the SARS-CoV-2 spike protein and its implications for immune escape and vaccine design. So the stage is all yours.
Thank you. Just give me a minute. Let me see whether I can share my screen. Okay, I'm sorry, it looks like some portion of the window may be cut. Can you hear me?
Yes, sir, you're perfectly audible and we can see your presentation as
well. Okay, fine. So let's get started with. So the title has already been read out, and so let me go straight away into the talk. From the title, the focus is on using bioinformatics methods at the level of sequence and the structure of the spike protein. And the bottom line is where we are looking at immune escape and trying to see whether we can predict the next sequence, which can be used as a potential vaccine. So as a brief introduction to the spike protein, this is the protein and this is the structure. On the top is a schematic view of the sequence and where it has domains. So it's made up of two major subunits, one called the S1 subunit, shown in red and gold over here, which has an N-terminal domain, a receptor binding domain, and it has the S2 subunit, which we're not too bothered about at the moment. It's involved with membrane fusion. So on the top right of the screen, which may be hidden, is the idea of why we are working on the spike protein. When you have the virus, the spike protein is on the surface, and therefore the only protein that is really visible to the host immune system. It binds to the host receptor as its primary aim, which is to get into the host cell, the ACE2 receptor. And therefore, the ACE2 receptor is this receptor binding domain. So that would be the initial focus of what we are looking at. Now, the structure has a sequence, and the sequence has these various domains. Now, the work on the SARS-CoV-2 over the last 3 years is basically encompassed or enveloped all of Indian and global science. And here are some 3 major papers which talk about this term immune escape. So they are in big journals, there's Science, there is PLOS Pathogens, and the title over here is essentially what I'm talking about. It's already about 2 years old. The SARS-CoV-2 variants, spike mutations, and immune escape. So if we already have this work published, why am I talking about it nearly 2 years later? So the reason I'm doing this is that there's an incredible amount of work that is being done on the spike protein, but our aim is not just to monitor and analyze existing data, but to try to use that particular data to predict how the sequence is evolving and predict the next sequence of SARS, the future sequence of SARS, so that we can prepare for it before it comes. So that's the real challenge, and that's the kind of predictive data science that we're using. So this is the protein. To summarize what we are trying to do as an objective of this talk, what are the residues responsible for the protein's stability and its fold, the protein's function, which is essentially what are the residues responsible for binding to the ACE2 receptor, and the protein and the residues that are recognized by antibodies and which need to be mutated to avoid the antibody response, and therefore what we call mutations responsible to escape the immune system, or the residues responsible for immune escape. Now, when we have this information, can we use it to predict the sequence of the next variant? For example, if we had alpha, beta, etc. strain, could we have predicted delta before it came on? If we have all this information up to Omicron, can we predict the next variant? Now, if we can do this, the sequence can be used as a vaccine for a future infection. For example, especially in the United States, not so much in countries like India, you have an annual influenza vaccine. So the strain that was used or available in the previous year is used as the vaccine. Now, what we are trying to do is to actually preempt this. Can you use the information that is available from previous strains to predict what will be the strain that's going to be the infectious agent of the next year, and therefore prime the population with that? Okay, so the first thing is, how do you predict the residues responsible for fold and for function? This is really from a little bit of dated work, 2 papers from my group, which is from 2010 and 2011 in PLOS One and in PLOS Computational Biology. So these are methods which can be used to predict a protein's fold and functional residues. Now, this is a schematic which shows you what I'm trying to talk about. On the top left is a protein structure. And I'm going to reduce the talk from a research level talk to something that may encompass even people who've come in from a background who just want to know what bioinformatics is, but there is a lot of research level content in it. So you'll have to please excuse me if I skip over some things. But very briefly, this is a structure of a protein, and the shape of the protein is responsible for being able to detect what it can bind to, which is the substrate. So the protein that is in consideration over here is something called a protein kinase. By varying residues around what we call the active site, you can change its recognition for a different substrate, but the overall protein shape remains the same. You also have a different kind of protein. This is called the Tim-Barrel structure, where you have the evolution of various active sites in different locations. Now, why am I showing you these structures? The structure shows you how the function of the protein is performed at the level of individual residues. But actually what we have is the information at the level of the sequence of residues. And this information at the sequence needs to be mapped to this structure. Now, what we can do is take a number of protein kinases. In this case, I've taken, for instance, 4 different families shown over here in 4 different panels. And they give you examples of the kind of residues. Now, you won't be able to see the residues exactly, so just look at the color coding. When you have a color that is common across all 4 families, now that is a protein residue in the protein that is conserved across all 4 families. However, if you have a substrate specificity determining residue, they would be conserved only in one subfamily and differently conserved in a different family. Okay, so what you have is you have a conservation in one subfamily and differently conserved in another family. So if you have this kind of data that is given to you, as human beings, we can look at it and visually find out by scanning through these various columns that I want to have this kind of a column that is conserved across the entire protein family and those that are conserved across subfamilies separately. And can I determine this? So these are methods that I'll discuss in the later stage, which I'll show you about. Now, what you have is that we can score this using these methods, and we have a score that can detect a protein that is a residue that is conserved across all of them. another score, which we'll call the RE function, which I'll describe in the course of the next few slides, which can be used to detect these. So you see when the RE function is higher than the RE fold, which is the pink color, then the residue is conserved differentially in one subfamily and not in the other. Similarly, if we have the Tim Battle kind of format, you have a conservation and you don't have anything necessarily conserved, or maybe even a gap in that particular area. So it's only conserved in one subfamily. Even in this case, the score that we have for the RE function will be higher than the fold, and those that are conserved otherwise. The other are sequences which are not really responsible and something that are space filling. So I'm only looking for, in the course of this sequence, I want to be able to score the protein residues which are responsible for the fold and then for the function. So how do I do this? So the first step is that we take all our sequences and we build it into a multiple sequence alignment, and then we use a model of the multiple sequence alignment called a hidden Markov model. And for this, we use a method from information theory called relative entropy, which can be used to detect the fold-specific residues. So this method simply scores the probability of occurrence of a particular residue in a particular column and divides it by the expected probability. So this is a relative entropy score, which is an information theoretic score. And for those of us who are not too hazy about mathematics, we can just simply remember that when this divided by this, when these two values are equal, this reduces to 1. So if the expected probability is the same as the observed probability, we get log of 1, and this tends towards 0. However, if this expected probability is much higher than the expected probability is much lower than the observed probability, we get a very high score over here. So when we get high scores for residues that are conserved, this score or this method can be used to be able to score columns which have residues which are highly conserved. Now, how do I get the other kind, which is the functional specific residue? There's a simple tweak of this equation. So instead of taking the entire probability of occurrence in one column, I take the probability of occurrence in one subfamily and then the probability of occurrence in the other subfamily. So I get two of them. Now, if we have this PIXY, such as this residue is the same as this residue, like I said earlier, the score reduces to 0. So it's no longer going to score residues that are conserved in both. But however, if a residue is conserved in one subfamily and differently conserved in another subfamily, this score is going to be very high. So you see the cumulative relative entropy score can be used to determine differentially conserved residues. So when we apply this together and I score each column together, it's called RE context. Essentially, we just use a statistical method to be able to combine and see which one is higher or lower. For example, if the CRE is greater than the RE, then this RE context is the CRE. Otherwise, the RE context is the negative of RE fold. So why do I put it as negative? Because the negative scores now come up at one level of the distribution. The positive scores come up at the other level of the distribution. And then we can just use a cutoff to be able to identify which residues are responsible for the fold and which residues are responsible for the function of this protein. And the other residues are not responsible for anything, responsible for either fold or for function. So now when I have my sequence of my protein and I stack it together with other structures and sequences of structures which are very similar to this, I will now be able to score these two and score each location on the sequence to be able to identify whether the residues is responsible for its fold or its function. So here is what we did with the spike protein. This is the raw data. This is a set of proteins from various organisms. Now, this is the clade of SARS-CoV-2. This is the SARS-CoV-1, which is the earlier pandemic. And these are other proteins which cause in various other animals the same kind of SARS-like infections. All of these bind to the ACE2 receptor. There are also other spike proteins from other viruses which do not bind to the spike 2 receptor. So what we have now is we have two multiple alignments, one corresponding to spike proteins, which bind to the ACE2 receptor, and the other spike proteins which do not bind to the ACE2 receptor. And by using those two methods that I had just described, we will be able to score and identify which of these residues which is conserved across this subfamily is either conserved in this or differently conserved in this subfamily. And these are the results. I think the slide is slightly offset, so we'll come back to it in the course of the sequence. So on the x-axis is the location on the receptor binding domain. There are approximately about 200 residues on it. Most residues don't have an involvement in either maintaining the overall fold or the function. A few of these residues shown in their location over here in the sequence and mapped to the location in the structure is highlighted in two different colors. So these are the residues which are receptor binding residues. Now, why are they important for us? Because if we design some sort of a monoclonal or a nanobody that can attach preferentially to this, we can block its function or binding to the ACE2 receptor. That's the first case, because these residues are forced to be invariant. If you have a loss of these residues or a mutation of these residues, then you lose ACE2 binding functions or binding to the receptor on those functions. So these are the most critical residues that are responsible on this receptor binding domain. Excuse me. Okay, so that's the first goal done. We can identify which residues are responsible for binding to ACE2 and which residues are responsible for maintaining this overall structure, both of which are critical for the overall functioning of this protein. Okay, so let's move to the next step. Now, the next step is not just simple sequence analysis of the protein. The next step is to use genomics. What is my aim now? My aim is now to look at all the sequences that we've had from alpha, beta, gamma, delta, and omicron. And there are millions of sequences. And to evolve some sort of a workflow, which I realized by listening into the previous session, you will have some sort of an understanding if you go through the courses and the tools that are provided by this platform. So we develop these ourselves and we run them on ourselves. So there's this data pre-processing, which is essentially what was called data wrangling in the earlier talk. So you take all this data, there are millions of sequences. We use a tool called NucMer, which takes the protein sequence of that particular section, maps it to the original sequence. And we have nuclear alignment, annotation of these, and you have the assessment of variants and the mutations. So these tools will map the variant or the mutation in a particular sequence compared to the wild type. And we have this information for the millions of sequences, each of which is tagged to a particular sequence, which is, you can now tag them as which variant they belong to. So this is the canonical data that is available from the WHO website. How do you define a various strain? So the alpha, beta, this is the technical terminology, B.1.1.7, etc. But let's use the Greek alphabets. So we have major strains, which are the alpha, beta, gamma, the delta, which caused a very big pandemic disturbance in India, and omicron, which is now the prevalent strain, which is post-vaccine, as well as newer variants of Omicron. And these are the various mutations that are used to define the variant. And these are canonical ones. So does our analysis, first of all, give us this, and what additional information do we get? Because these are just canonical ones that are described by WHO. So by using this method on these millions of sequences, we can map them out. The graph over here shows the spike protein sequence, which is in this is the nucleotide sequence. So the numbering is a little different, but it's originally around 1,200 odd residues. And the location at which this mutation is present, and when it's right up to the top, which is at 100%, the entire strain has these mutations at 100%. But we're not just looking at the 100% because that's used to define the mutation. We're also looking at those which have come up and also be able to map them out in terms of time and the location that they are present. Now, one of the things that you could look at this graph is that alpha, beta, gamma, and delta show a natural progression in the evolution of these strains. They don't necessarily move from alpha to beta, etc. They could have moved from the wild type through various strains before they became dominant strains. But this did not have any immune response to them. Now, after Delta, most of India, for example, was vaccinated. And therefore, the Omicron strain that came up is a post-vaccine strain. So most of these mutations would have had to occur to escape the antibody response to the wild type protein. Okay? Question, or is there someone whose mic is on? All right. Okay, so essentially what we do is monitoring and mapping out this data is the first stage, and being able to put it down onto the structure also shows us these various locations. So if you remember from the earlier schematic, this is the location of the various domains on the sequence. The important one is the receptor binding domain. Also important for now for immune escape is the N-terminal domain, which is visible on the surface over here. So when we map this, you can see most of them are really white and therefore not too important, but there are some residues which are responsible and which are colored up over here and which are found. And as you can see also from this, there are very few with alpha, few with beta, few. There are much, much more with Omicron, which had to mutate extensively to be able to become a dominant strain, since most of the population in the host that it was infecting had been immunized. So this also gives us two types of mutations. When you look at the mutations at the top, which are exposed to the immune system, these have a selective pressure. These don't have a selective pressure because they are not exposed to the immune system. They are embedded within the membrane. So we can also use this information, which I'll describe later in the talk, to be able to identify how to get positive selection for immune escape. So these are all mutations that are present on the protein, which are involved with having to detect a new antibody. Now just look at the schematic, because if you want to get, we need a mathematical model, but if you like visualizing data, here's it on a structure, which is what we've been doing now for 50 odd years. What happens is that when you have the wild type protein, which looks like this ice cream in white, in vanilla, you have mutations in the alpha first, which come up, which don't cause too much of a disturbance. And then you have more mutations that are coming in. Now you see that a number of mutations on this N-terminal from the beta are also present in Omicron. The Delta was a far more virulent strain, and it had a few mutations which could have been responsible for virulence, not the focus of my talk, but these are the mutations that have come up on top of these visualizations. Now, all of these would be small mutations that are present in locations on this sequence, okay? Now, what I'd like to do is use this information to be able to predict on the sequence what would be the next residue in what location on this protein, so that I can get the next strain from Omicron, all right? So this is the second part of what we've done, where you use genomics information, and you'll be able to identify from this genomics information what is the mutation rate, what is the type of mutation, and what are the residues that are present on the protein. So once we've finished with this, the data that we get, which is the mutation information, we can do two types of work with it. The first is, if you take a mutation table along with the frequencies separately for each of these variants, and you use the structure of each of these variants, we can map these mutations and predict spike epitopes. We use a tool called Discotope from the structure. So from the structure, if you use this tool Discotope, you can predict what will be the epitopes on the structure and overlay the variant mutations on that to identify their particular rate of immune escape. So here's a bit of it. I'll read it out. Actually, I'll leave it out, maybe for some, in the interest of time, because it's simply some information about what we have as the SARS-CoV-2 spike protein. It's exposed at the surface, and it is key to infection. Now, what we're really interested at this stage of the talk is that an epitope, which is known as the antigenic determinant, is the part of the antigen that is recognized by the immune system, specifically by antibodies from B cells or even by T cells. Now, the T cell epitope prediction is slightly different, but you can integrate it into the method, because what happens is that the protein is sometimes digested and there are smaller parts of the protein that are exposed onto the surface of antigen-presenting cells. But the B cell and antibody recognition is on the primary protein. So the epitope can be determined directly from the protein structure. So when we identify these B cell and T cell epitopes on the protein, and then we map these mutations, we can see which mutations are going to can be detected and overlay on that block the antibody response against the wild type protein, that overcome the antibody response against the wild type protein. The antibody that was generated from the wild type protein can no longer recognize the mutated protein if it is mutated in those locations. Now, this is the result of the antigen epitope. To describe the result, on the x-axis is the location of the sequence. There are approximately about 1,200 residues for the spike protein. And you have a score for the epitope propensity in a location on the residue. And what we have mapped over here is an overlay of the mutations that overlay with the immune escape. So you have on the wild type, this has colored up mutations across all of them. Why this slide is important with the coloring is to show you that Omicron has lost an area that is present over here around 180, position number 180. There's a loop that disappeared in the overall structure. And that is why one of the principal reasons why the antibody response that was prepared from the wild type, which was used as the original vaccine, no longer has much of an effect on the Omicron. So you see, there's a very, very strong correlation shown over here with these red lines between where we have mutations and where we have areas which are epitopes. Now, why is this important? From a data science perspective, I don't need to look for mutations elsewhere. They may be responsible for other things such as virulence, susceptibility, transmission, etc. But for binding and for recognition by the immune system, these are the residues that are critical. So we need to focus only on this, and then we'll be able to detect locations and residues which can be responsible for immune escape. So this is a result which is a different way of showing what was earlier presented. This is the actual data that is present in the Omicron, which was shown in an earlier slide, and it shows you the overlay with the earlier slide with the structures. Now, there could be a mapping from this. The coloring is not very easy to make out, so let me just describe this. What we have is with the Omicron protein, the sequence shown over here will map to the various domains. So for instance, if I'm looking for the N-terminal domain, I'm looking at the location from about 13 to 305. And for the receptor binding domain from this region. Now you will see that there's a region over here, which is towards the end of the receptor binding domain, which is highly epitopic. I mean, antigenic, which has a large number of epitopes, and so is the region that is just below it. So this is the region that we need to focus on, and they are colored up over here from the mutation rate. So Omicron has mutations in most of these regions that are mapped, as you can see, 339, 348, etc. Most of these regions, from 400, 484, these regions that are responsible for immune escape. So what we've got so far is that you take a classical sequence analysis method by strains from various organisms of the spike protein, and you overlay them and you understand the protein structure. Then you take the sequences of the SARS-CoV-2 itself in the last pandemic, millions of them, and again identify mutations using standard genomics methods and using standard analysis methods. Now, once you have that kind of a data, you can also use an evolutionary analysis, because what you have is mutations that are happening. The tool that is commonly used over here is HiFi, And you can identify sites which undergo positive and negative selection using what they call fixed effects likelihood. And both of these will correspond to what we will call significant sites. Now, here is the example of what we are talking about. Essentially, a non-synonymous mutation, which is a mutation which changes the amino acid. It directly affects protein function, which influences the fitness of an organism. Mutations that leave the amino acid sequence unchanged are synonymous mutations. Now, under negative or purifying selection, less fit non-synonymous mutations accumulate more than synonymous mutations. So essentially, if you have negative or purifying selection, what we call purifying selection, you will have more of the non-synonymous mutations and then the synonymous mutations. However, when you have positive selection or diversifying selection, which forces the protein to mutate far more, then the inverse is true. So what we need to do is have a method which can monitor a rate of non-synonymous substitutions and synonymous substitutions. And there is a score that is made by the program HiFi. It's well known in the theory for now nearly 40 years. And you have a ratio, which is one of these, the non-synonymous mutation divided by the synonymous mutation. And it's a standard measure for selection. So there are two types of selection you can classify over here, the positive selection or the negative selection. And if there's both of them are equal, if the rate is the same, then it's neutral evolution. Okay. So if we take all this data together and we qualify the kind of mutations that we are getting into, non-synonymous mutations and synonymous mutation, or rather purifying and diversifying mutations, we can see that we have a mapping of the kind. Now we've reduced the kind of mutations that are present in the spike protein to a much fewer number that are responsible for the case that we are looking at, which is for immune escape. Okay, so this is where we were. Now let's move to the last stage of what we want to do, because it's not just mapping and identifying residues. We want to use this information to be able to predict what's happening. Now, the classical ways of doing data science would be to build a classifier, because if I have this kind of variant information, I can build a classifier. So give me a sequence and I'll be able to tell you what strain it belongs to by building a classifier based on the variations that are present over there. A second type of classifier would be able to predict sites responsible for immune escape, both from this kind of information. Now, what we'd like to do is look at forecasting the next level of sequence, which is by regression. Now, this is not easy to do. The reason is that the longer a protein, the more the number of potential sequences that can be generated by this. This is just a theoretical exercise to tell you this, because each location can have 20 different amino acids, and therefore you have this many number of combinations. But this is not really true. Why? Because in the past methods that we've used, we first of all reduced this to a number of sites. It's not just the overall protein length that we are interested in. And we've also reduced this to what type of amino acids are available and allowed. And the earlier residues that were already used cannot be reused. So we have methods that we can use. Now, when we are dealing with this in terms of time, you can't necessarily pick up the residues because substitution and reversion is now hidden from your data. But now we have time-based data. So we have both this residue and we can map out both substitution as well as reversion. So what we'd like to do is build a model which we can convert. And in this case, we can convert it into a continuous regression model. So this tool has been developed and available on GitHub by Eric, Eric ML. It's the link is present over here. It was used for influenza, and we modified it slightly for using with the SARS-CoV-2. So this is essentially what happens. Just as everyone in the world now knows about ChatGPT, ChatGPT is a large language model, which is called a generative AI model. So what we are dealing with over here is also a generative AI model, but instead of dealing it with large languages, we've fed in the previous information present on the sequence evolution and trying to ask whether it can predict the next sequence. And this is known as a variational autoencoder. So you have a discrete to continuous conversion. and you have some sort of a scaling that is done. This is a schematic, and this is the code. So essentially, variational autoencoders are unsupervised learning techniques which leverage neural networks for representational learning. Now, there are specific type of feed-forward neural networks where the input is the same as the output. So you have two types, which is three components. The first is the encoder, the second is code, and the third is the decoder. So you have a hidden layer that describes the code used to represent the input, and the network may be viewed as consisting of just two parts, which is the encoder function and the decoder function, which produces a reconstruction from that particular code. Now, in some ways, you will have to read up, and we'll have to come before we start using this at the theory level to be able to pick up on machine learning, to get to artificial neural networks, etc. I will skip over that, and let's just use this as a black box. Okay, essentially what we need to do is put in the information on the variation, and we get this code. So how do we implement this if we are given something like code that is here? So you take your viral sequences, and the first stage is to be able to convert this data, which basically has a collection date, and you have a protein sequence that is generated from that, into some sort of a binary format. So we convert it into a binary format, and this is now translated from the discrete form into a continuous representation in lower dimensions. So what you have now is, you can visualize this more or less like a flowing graph, the continuous representation. And the way to model this is to use what is known as a Gaussian process regression, which forecasts future continuous coordinates. So given some sort of a regression line, a continuous regression line, I can predict what the next value would be. So what is this idea of the Gaussian process? The basic assumption is that if you say two vectors, let's say x and x dash, are similar, then their functions, fx and fx dash, should also be similar. So the covariance function between these two will give you a measure of similarity between these two vectors, x and x dash, and the mean function mx encodes the a priori expectation of the unknown function. So this unknown function values are conditioned using the known ones. If there's no similar values, then the mean function influences the result. So translated into biological English, I have sequences of the virus so far, and those are the known values that I want to use. Can I massage them into this particular model such that we can predict the next sequence that is coming out? Okay, so once we've got this data that is fed in over here, the variable autoencoder decoder now constructs new sequences from these forecasted coordinates. So now I'm getting new sequences which are predicted based on the timelines and based on the kind of progressions that have come in from the earlier sequence. And we can then synthesize these sequences and see whether, and rank them and see what, there is an internal likelihood measure, but we can also use other measures that we've described earlier, such as are the location of the residues of importance in the locations we are looking at for immune escape. So this kind of model forecasts the held out sequences. So these are some of the results that we have. Essentially, this is the sequence, and shown in red over here are the forecast points, which fit very well with what is expected. Okay, so when we put all this data back in, we can see which residues are predicted to change. There are a number of residues which could be false positive, but they are filtered out from our information earlier. So the ones that are in the locations that we are looking at and which we have defined as important, those are the residues that we can use to filter out from these 70 odd sequences that were generated in this exercise, and predict which sequences would be the sequences which be of relevance. Okay, so this is my end. I think we've reached the timeline also, a little over time, I'm sorry. So you implement analytical methods which are responsible for protein stability and fold, the protein function, and immune escape, and we use this information with generative AI to predict the sequence of future sequence of the spike proteins. And this can be used to generate a vaccine which will predict future infection. Okay, my apologies for running short on time. I didn't have the clock present over here. So thank you. I hand back to the chair.
Thank you so much, Dr. Andrew Flynn, for that wonderful presentation. So we do have some queries in the chat that I would like to quickly take up. I apologize to Dr. Gupta. We'll be starting with the session in just a few minutes. So the one question that we have is, what does gap indicate in protein structure? Will it be the void space or is it the sequence?
Okay, is my sharing still on? I'm sorry, I really don't know how to stop this.
We can see you.
Okay, I'm trying to switch it off. 1 minute. Okay, very briefly, a gap in a multiple alignment is what we call either an insertion in some sequences or a deletion in subsequences. In bioinformatics, it's called an indel. So when you stack together these sequences, if there is a deletion in a few sequences, the other sequences have the sequence which is an insertion present, and it will the sequences will come out as a gap. Okay, so it is only present in a few sequences and absent in the other sequences.
Much. And then the next question is, were there any unexpected findings in your research, sir?
Okay, I'm just listening to you because I can't see the chat. You want me to read it from the chat? So just repeat the question.
Yes, sir. Were there any unexpected findings in your research work?
No, because we were looking for a particular question. We used a standard method to be able to arrive at that answer. So we weren't really getting anything unexpected, if that's the thing. We focused our particular research on a hypothesis, and we arrived at that particular result. So the answer is no.
Thank you so much. So we do have some more queries. We'll be sharing it with you during the panel discussion, or we'll mail it to you. So we look forward to hearing.
Yes, I apologize for taking so much of time. Okay, take care.
Problem. It was a very wonderful presentation. So with that, now I'd like to invite Dr. Mukesh Gupta. So let me give you a brief. Dr. Gupta is a fellow of the Royal Society of Biology. He earned his PhD in animal biotechnology from Punjab University. He started his research career as an associate professor of animal biotechnology at Punjab University in 2007. He's currently working as a professor of biotechnology and medical engineering at the National Institute of Technology, Roorkela. He is also the scientific steering committee member of ARPS. To share insights on omics of spermatozoa and its relevance to male fertility, I'm most honored to invite Dr. Gupta to start with the session. Over to you, sir.
Thank you. Thank you for the inviting. First of all, thank you for inviting for this symposium. And I'm very happy to share my research data with the scientific community. Let me first share the slides. Is my slides visible to all?
Yes, sir. It is not in slideshow. Yes, now it is. Thank you.
Yes. So I'm thank you once again for inviting and giving me this opportunity to share our data. I'm going to completely move from the in silico analysis and prediction method to utilizing those technology for understanding something which was hitherto unknown to us. So I'll be presenting talking about both wet lab data as well as the prediction data that we have done in our laboratory. What we are going to talk is understanding the spermatozoa function and its relevance to fertility from the omics point of view. Perhaps many of you will be aware that the infertility has been becoming a very common phenomena across the countries, particularly the developed countries and countries with the rapid urbanization. Globally, we believe that 60 to 80 millions of couples are infertile worldwide. In India alone, estimated to have 15 to 20 million couples infertile. In many countries, it is generally believed that the cause of infertility or inability to bear the baby lies with the female, which is in sharp contrast to what science says, is that 50% of infertility cases arise from the male factor infertility. And 1 in every 15 to 20 men are diagnosed to be infertile worldwide. This data largely comes from the urban area, but perhaps similar things exist in the rural areas as well. Now, the cause of fertility could be a variety of them. There could be disorders of the semen. There could be abnormal sperm motility. There could be asthenozoospermia, oligozoospermia being very, very common. There could be the failures of testes, including cryptorchism or the orchitis or varicocele, and could be many. But there are roughly 20% of the infertility cases where we are unable to find the cause of fertility. Simply the sperms are infertile. Say, for example, the spermatogenic failure, where there is no sperm production at all. So fertility cause could be a variety. And so far, we have been trying to understand this from semen analysis or hormonal analysis or the diagnostic tools such as ultrasound and so on. In very recent terms, we are trying to understand molecular point of view. Now, from that, we have in our laboratory started investigating that what other factors could be responsible for the sperm fertility. Now, sperm traditionally is considered as just a delivery boy. It is considered that the job of the sperm is only to deliver the nucleus into the cytoplasm of oocyte, and which shall participate to form the pronucleus and to form the embryo. But with time, we get to know the job of the sperm is not just to deliver the pattern of DNA or the nucleus, but also it takes part in formation of centriole and other organelles which can go, particularly centriole. On the other hand, certain organs, if delivered, they could be dangerous to the embryotoxic. Say, for example, the mitochondria, which are in the middle piece of spermatozoa. If they delivered into the cytoplasm, it may lead to mitochondrial heteroplasmy and embryonic death. We also came to know with time that there could be soluble factors present in the cytoplasm, which might help in oocyte activation, triggering the resumption of meiosis in oocytes and the cell signaling, particularly calcium signaling, so that the cells start dividing the embryonic cells to become the embryonic cells. And with that, most of the factors limited, most of the research limited to that sperm function, and there we stop. Now, in our laboratories and some other few laboratories, particularly from Pennsylvania, we have reported that the sperm also contains a very, very tiny amount of spermatozoa. It's estimated to be about 0.015 picogram of RNA, which is very, very tiny if you consider the per cell concentration. But if you assume the volume of cytoplasm that is sperm has largely nucleus, very tiny amount of cytoplasm, then if you think from that perspective, this RNA quantity is pretty high. And it was discovered at the time that earlier people trying to evaluate one or few RNA in spermatozoa, one at a time, a few at a time. And traditional methods of evaluating RNA population in spermatozoa did not give us much information. With the invent of omics technologies, particularly after the Illumina sequencing came into picture, then we, and along with others, have identified that there could be more than 3,500 types of RNAs which are present, and some of them could be total length, full length RNAs that are present in this spermatozoa, that include coding sequences, coding mRNAs, and non-coding sequences, including miRNA, piRNA, circular RNAs, and long non-coding RNAs. When although this was very paradoxical, because it is in the traditional methods, we say that this is just residual. They're not supposed to be present. They are present by virtue of being a reminiscent of spermatogenesis. And why this thought was there that nuclear genome in spermatozoa is transcriptionally dormant, except for mitochondrial DNA, where they say probably some might function, some might be synthesized. And then the spermatozoa have abundant rRNA, including 20S, 28S, and 18S rRNA. Therefore, there is absolutely no possibility of translation. There is complete translation shutdown. There is no de novo protein synthesis. So the data which was in paradox, that the omics technology has given a data that there could be mRNA. It was initially thought that perhaps this could be a contaminant from the somatic cells or the other cells that might get mixed up with the sperm samples. Then there are a number of methods that was employed to get a purified population of spermatozoa, and it was confirmed that these are not just the errors of the sequencing, not the errors of Illumina sequencing. They are indeed present. And with parallelly, when people try to do the traditional full-length cDNA library synthesis, then they get to know, yes, the spermatozoa also contains some RNAs which are in full-length sequence. Then the question comes, what is their source and what is their function? Now, there could be a variety of the function. Literature showed that if you look at this, the presence of the spermatozoal RNA, when they try to co-localize, the spermatozoa has a very peculiar location of RNAs. So the nucleosomes of the sperm is highly compact with a solenoid structure. The DNA is bound with highly basic protein, which is glutamine, unlike the somatic cells where we have the histone bound DNA, where we have glutamine bound DNA, and they take a solenoid structure. So very difficult for RNAs to transcribe. But then there were few, particularly in those near the nuclear matrix area, there were few amounts of the histone proteins to which DNAs were bound, and it was then thought that perhaps those RNA which are present here at the bound to histones are probably potentiated to translate to a protein. But so not many hypotheses put in, but nothing was coming in reality. What would be the reason? So we tried to investigate that first with some wet lab experiments. We took the sperm samples of 3 different pigs, three bows, 1, 2, 3, which were known to have different ability to develop into blastocyst or the embryonic development. This is low fertility, this is high fertility, and this is medium fertility. And we stained the spermatozoa with the RNA-specific dye, the dye which can bind exclusively to spermatozoa. And what we could observe was that the percentage of RNA positive, the RNAs are very tiny, so very difficult to obtain the positive signals. But whatever we could obtain, we could see that there was a positive correlation between the number of RNA positive spermatozoa with the blastocyst rate. So as the percentage of such cells increased, the percentage of spermatozoa, or the percentage of blastocyst seems to be increasing. So it does suggested that perhaps the spermatozoal RNA in the spermatozoa may have some function to do that. So before venturing into detailed analysis, what we did is we first took the spermatozoal RNA, we purified them and injected them into oocytes, pig oocytes, freshly collected oocytes, and tried to see if this spermatozoal RNA, if injected, can develop into embryo, which we found that they couldn't develop. So we started using the oocytes, injected them the spermatozoal RNA, and employed an electrical signal to activate the embryonic activation, embryonic development. So where we could find the embryonic development. So what we found is, very interestingly, the parthenogenetic activation, which is without spermatozoa, they can develop to blastocyst stage. But if we inject the spermatozoal RNA and then activate, the rate of embryonic development increases. And not just that, the embryonic development the embryo quality that we got was better in terms of the cell number, in terms of the apoptosis and all, which I'm not showing the data. They were much better compared to the normal parthenogenetic embryo. So we know that, okay, that means this spermatozoal RNA has perhaps some function. The next set of experiment, what we did, we used a new method to deplete those RNA. So we used the spermatozoa, depleted them with the spermatozoal RNA, and then used them for IVF, in vitro fertilization of pig oocytes. What we found was, when we depleted the spermatozoa, very few embryos could develop. You can see roughly 6%. Majority of them could not develop. That means it reinforces the idea that spermatozoal RNA have some function, because when we deplete them, it reduces development. When you increase them, it improves the development. So we tried to do a rescue experiment. What we did, we took the spermatozoa, depleted the RNA from them, injected, and then we used that RNA depleted spermatozoa for IVF. And after fertilization, we injected spermatozoal RNA into those eggs. And we found that it could rescue the development. So if you see, this is the normal IVF, 31% embryo develop up to blastocyst stage. Whereas when we depleted the spermatozoa, it dropped down to 5%. And when we injected spermatozoal RNA, it gained back to 21%. That means spermatozoal RNA does have some function. So next approach, then we took to try to understand what could be the reason. If there are RNAs, there could be two possibilities. Either the non-coding RNAs must cause downregulation of some gene related to embryonic development, or there could be coding RNA which gets translated into embryos, and they may either downregulate or upregulate depending on whether they are regulatory in nature or they are proteins of the some signaling pathway. So keeping in this view, what we did, we took spermatozoa, one in which we injected the spermatozoal RNA and then parthenogenetic activation, and one without spermatozoal RNA, and we compared them with the normal in vitro fertilization. So we took 3 groups of embryos and employed the LC-MS to do a global peptide sequencing. We did global peptide sequencing, and we also applied EQUA for absolute quantification of some of the selected proteins. So what we found, we could actually get about 54,000 spectra. And these proteins could map to about, these spectra could map to about the 1353 proteins. And if you look at that, the parthenogenetic embryo, the green ones are the one which is injected with RNAs, and the blue ones are those which are injected with the IVF. We could find that there were many proteins which were common, but then each one has their individual unique proteins present in them. And if you look at the concentration wise, when we normalize the protein set with the parthenogenetic activated embryo, we could find that in the RNA group as well as the IVF group, a large number of protein got upregulated by more than 10-fold. So in RNA group, 228 protein got upregulated, and in IVF group, 93 got upregulated. That means probably some of the RNAs are getting translated to protein in the eggs after the injection. And many proteins could get downregulated also. In fact, number of proteins which got downregulated were much higher than those which got upregulated. So perhaps there are also regulatory sequences like mRNA sequences present in the spermatozoa. So there were also the unique proteins we got. Say if you compare the RNA group, RNA injected group versus IVF group, both group contains 50 proteins which were common, and there were 42 proteins unique to IVF group, and 152 proteins were unique, new in RNA group. Similarly, about 39 were absent in the RNA group alone compared to parthenogenetic, and 266 were absent in IVF group compared to parthenogenetic embryo. So our hypothesis was, and these proteins, when you look at, they belong to a different cellular mechanism, a great majority of which were involved in RNA metabolism or DNA metabolism or protein metabolism or some of the base cell metabolism. But a good number was also involved in the signal transduction pathway and different other physiological processes. So this suggests, what we understand is, if we find the proteins which are upregulated or downregulated, there could be two reasons. If we have a change in both the groups, that means the proteins have some role to play during embryonic development. If we have only in one group, and if that is due to RNA group, that means that RNAs are probably those which have function during the spermatogenesis, and those which are in IVF groups are the one which probably have function during the embryonic development. So this supports the basic hypothesis that the RNAs which were identified by the Illumina sequencings were not the errors. The RNAs does seem to have some function. So then we switched on to another species, goat, and using as a model system, we have a very good model system for spermatogenesis. And then what we did, we started a detailed analysis of RNA population in goat, and then investigating whether do they have any function. So we employed 3 different methods to sequence all the RNA population that are present in spermatozoa. We employed RLM race and constructed a full-length cDNA library. We employed short-read Illumina sequencing method, and we also employed the long-read nanopore sequencing. The RLM race and nanopore sequencing we have in our laboratory. We employed that. Illumina sequencing, we outsourced it to another company. We got the data, we analyzed them, we combined them, and then we employed normal process, which we all know. We trimmed the data sets, mapped them to the databases, and then try to calculate the FPKM value and do gene ontology analysis for the biological function or cytoplasmic location or molecular function, then the pathway analysis. We also built up the PPI network, protein-protein interaction network, and try to analyze those network where they could participate. Now, what we could that, in all the 3 methods, we could find that there are full-length sequences, both in Illumina, nanopore, as well as the race. There were some sequences we could obtain the complete RNA sequence, starting from the first exon to the last exon. And some of these were involved in the male reproduction, says androgen receptor, cat spur, which is important for the capacitation event, the cat spur, which is an ion channel for capacitation. Dazzle, very good marker for spermatogenesis. Heat shock protein, SPCA, protamine, very important for the compaction of the sperm nucleus. And 3 sequences, all the sequences had some of the genes which were common to all of them. That means we could infer from that genes our sequencing are working. So as expected, we could find that the majority of these sequences belong to the mRNAs, but it also included the microRNAs, and lot many the rRNAs. Of course, rRNAs, we did riboreduction, so numbers were pretty less. Otherwise, they could have been more. We did get some rRNA despite riboreduction. And then we also predicted some of the circular RNAs and long non-coding RNAs. If you look at this graph here, we predicted, and show you later, we developed a novel pipeline to identify the circular RNA, and we could find number of circular RNAs within the spermatozoa. The gene set, interestingly, also included about 160 imprinted genes. Probably all of you are aware the imprinted genes are those genes which are expressed from uniparental gene, either from the paternal chromosome or from the maternal chromosome. So out of 160, which are located in 29 different chromosomes, 79 were maternal and 81 were paternally imprinted genes, we could predict. And there were also many long non-coding RNAs, 591 we predicted, and we also predicted their targets. And many of the target genes were known to be participating in one or the other steps of the male reproduction. And not many of them were for the spermatogenesis process. Some of them were also involved in the early embryonic development. And these proteins, when you try to do the gene ontology, they were belonging to several pathways. Some important pathways, which later we also discovered was, there are many were involved in the Wnt signaling pathway, MAP kinase pathway, and AMPK pathways, and TNF-alpha. These TNF tumor necrotic factor pathways. They were, of course, their localizations were there to many of the other proteins. And when you try to cluster them for those genes which are fertility, known to be male fertility related, we could also see the involvement of not many genes in the GPCR pathways, G protein coupled receptor signaling pathways. Some of them were also involved in the early embryonic development or neonatal development, including the formation of the brain or limbic system. But there were a good number of proteins. So it probably suggests, these analyses suggest that sperm RNA could have function during fertilization. That means could be either capacitation, I'll come to that later. And the mRNAs may remain in the cytoplasm to affect some signaling pathway post-fertilization that may play a role in the early embryonic development or the postnatal development. So those signaling pathways may be affected by them. So we took up the first one, whether they affect the sperm function itself. So if you know, probably some of you know that when the sperm has to function, sperm has to undergo a process of capacitation, during which they acquire the ability to fertilize the egg. So we started doing it. We collected the spermatozoa, purified it, the motile spermatozoa, and we treated them with the capacitating agent. One group we kept in a non-capacitating media, and then other one capacitating media with extra added heparin, which improves the fertility, improves the sperm motility, hypermotility, and capacitation. Then we isolated the mRNA from them, constructed the cDNA library and did an Illumina sequencing and mapped them to genome and tried to understand the function of whether this spermatozoal RNA plays any role in the spermatozoal capacitation. So as you can see from there, when we treat the spermatozoa with the heparin, they improves the motility and capacitation. So within 4 hours, we can see there is many increased number. Nearly 80% sperm become capacitated and shown autosomal reaction, which is a mark of that the capacitation has taken place, an essential requirement for fertilization to take place. So we could see that heparin can cause the capacitation and autosomal reaction, which are requirements for sperm to fertilize. Then we did the RNA sequencing of them, both the groups. We found that while many mRNA populations were common, there were many of the genes which are upregulated in the heparin treated or the capacitated spermatozoa. The fold changes were not much, which was anticipated because considering the very tiny amount of spermatozoa we have. But then we had about 96 proteins which were shown the fold change of between 5 to 10. If you look at this, most proteins were upregulated. Very few were downregulated genes. Majority of them were upregulated. If I show you the volcano plot, you can see these differences. Many proteins were upregulated and very tiny fractions were reduced in the expression level of the genes. So sperm RNA have when we expose them to capacitating environment, many of the sperm RNA numbers increases. Now the question of course remains, which I shall discuss later, where from they are coming. But then there is definitely an increase in the RNA population once you activate them. And if you look at this, the capacitation induced proteins in the biological processes, they involve in many molecular functions, including the DNA binding. They had many DNA binding RNAs, and metal ion binding, serine, threonine kinase activity. Then some of them were ribosomal, the ATP binding. There are many binding proteins. The one which we had the upregulated ones. And when we try to analyze them, we find there are many genes which are involved in, as we have seen earlier, signaling pathways known to be related to capacitation, like AMPK pathway, calcium signaling pathway, cyclic AMP pathway, cyclic GMP pathway, MAP kinase pathway, phospholipid PLD pathway, PIK3 AKT pathway. So many genes were involved in them. And these genes, which I've listed here, are earlier known to have impact on capacitation on one gene at a time basis analysis. And we verified, we selected some of the data, some of the samples. We did the real-time RT-PCR, reverse transcriptase polymerase reaction, and verified that the data which we generated from the Illumina sequencings are in line with those which get with the RT-PCR or real-time PCR. So among these pathways, we saw was many of them were also involved in the ROS activity, oxidative stress pathway. So we then tried to see if really there was an increase in the oxidation or the reactive oxygen species. And we found that when there is a capacitation event, the activity of the reactive oxygen species increases. You can see that more number of green cells, which means that greater number of cells were having the high ROS activity, and it was parallelly matching with the percentage of cells that they got capacitated. So by 4 hours, many of them showed the ROS positive cells, very much increased cells. So it then reconfirms our idea from the in silico, from the omics data, that the oxidative stress pathway was indeed activated in the spermatozoa. So we tried to correlate all the data and look at the capacitation pathway. And if you look at our proposed model based on these data analysis is that heparin, it increases the ROS activity and opens up the cat's paw, to which the calcium goes in. And this heparin can also bind with the BSP receptor to activate the soluble acetyl cyclase enzymes in the cytoplasm. And of course, also cause the sodium bicarbonate channel to open, which all together increases the activity of cyclic AMP. These are the genes which are involved in this pathway, the star ones, we have seen them. So this, the cyclic AMP then increases the pKa, which together with the AKP increase the protein tyrosine kinases to activate the events of the tyrosine phosphorylation to somehow cause the hyperactivation and capacitation event. The oxidative phosphorylation events also participate in the same. So the one which we are seeing the star marked, we have experimentally verified to confirm our data with the omics data. So this is the pathway we find with the heparin. But then I'm not discussing today, but we also try to change the capacitating agent from heparin to another molecule called arginine. L-arginine, also known to cause in our data increase the percentage of capacitated spermatozoa. So we also did that. We took the spermatozoa and exposed them to the non-capacitating medium or arginine, and we used another molecule called PHE, penicillamine hypotaurin epinephrine cocktail, and then analyzed the RNA sequences within them. to find out differentially expressed gene and then try to see the pathway. We could see that they're also increasing the capacitation, although the pathway is a bit different. Nonetheless, all of them follows the oxidative phosphorylation pathway. Say, for example, in case of arginine, they produce nitric oxide, which can then activate the acetyl cyclase, and they can also participate in the RAS and activate the guanylyl cyclase activity to cause the serine-threonine phosphorylation, tyrosine phosphorylation, and increase in MAP kinase activity. So all these proteins, mRNAs, which are participating in these signaling pathways were known. Similar data we also got with the penicillin, epinephrine, and hypotaurine. So if I sum up together these data, our data suggests that unlike traditional methods of investigating on one or few RNAs, the question whether the spermatozoal RNA were reminiscent of spermatogenesis or did we really get them, it opened up the whole plethora of a whole new suite of RNAs, including circular RNA, long non-coding RNAs, and many of the microRNAs being present within them. Our data also said that these spermatozoa, when we inject, they improve the blastocyst formation and the global protein profiling. We have actually also done the absolute quantification of this protein, but due to paucity of time, I'm not discussing on them. Then we also saw that when depleted, it inhibited the embryonic development. And when we try to inject the sperm RNA into these depleted spermatozoa, there's a rescue of early embryonic development. So these data suggest the spermatozoal RNAs are not residual of spermatogenesis. They do have a function in early embryonic development, and they also have function in the capacitation events, at least the one which you have analyzed. Further activities are going on. Now, I also give you a very brief comparison of when you try to apply a full-length cDNA library reconstruction by race versus Illumina in nanopore. Not much work has been done with the nanopore sequencing, and considering the samples such as spermatozoa, which is very, very difficult to isolate the sperm. What we found that with the nanopore sequencing and RLM rays, we can get the full-length information, which is hitherto not possible to get good with the Illumina, but we got in nanopore sequencing the around 1.5 kb length. In some cases, we got up to 3 kb also. But in case of Illumina, we get, of course, as we know, it's a more number of reads. So if you want to looking for the global profiling, Illumina seems to be good. But if you want to know more about the analysis about what kind of transcripts are present with function, in cases like sperm, it is questionable whether these genes are really reminiscent of functionals. But if full-length information you have, then you know that these genes are bound to have some function. So in that angle, Nanopore seems to be good. We have also developed I didn't discuss about this pipeline for circular RNA and LNC RNA, but they seems to be involved in different stages of spermatogenesis and embryonic development. And we have shown that the spermatozoa participate in Wnt signaling, CMP pathway, MAP kinase pathway, signaling pathway, including oxidative stress pathway. And when we did this staining and confirmed that the capacitation in stages involves ROS activity, which correlates with the omics data, suggesting that the capacitation process involves some of the upregulation of the RNAs and thereby causing the capacitation. So it gives us a lot of information which was earlier unknown. I have not cited the references. All this data has been published recently in SCI journals. And that's all for my presentation. I must acknowledge the funding agencies before I close. The GBT, the ICMR, the CSIR. CSIR has given the maximum fund for this purpose. And validation studies and all come from the ICMR. The in silico studies comes from the DBT, and also the SERV, which has been funding us. And of course, we should not undermine our students, both undergraduate, postgraduate, and PhD students who have been the backbone to provide this data. Thank you for listening.
Thank you so much, Dr. Gupta, for that wonderful presentation. But due to time constraints, I'm going to have to limit the number of participants' queries that I have to share with you. So the one query that we have is, can you suggest any open source or publicly available data sets or tools that you would recommend for me to start working on this area? Sir, you are on mute at the moment.
So if you are looking for the data sets for analyzing species like human mouse, then open source data sets are also available, and there are tools which are available. But if you're working in other mammalian species, farm animals like pigs or goats, where the database remains incomplete, so in those use cases, you have to write your own code. Our codes are available in the GitHub. You can get the source code from there. And we have also developed some of the pipeline by circular RNA, which you can get from the GitHub. If you search for my name, you'll get there. And data sets which we are having, we are still performing some analysis, so we have not yet uploaded in the Geneomnibus GEO site. So we'll upload it shortly because we are still doing. This is the novel study so far. Other than us, there's no study on proteomic study. Nobody has done because it's not easy to do that. You need thousands of eggs, fertilized egg, to do that. It's very difficult. So proteomic study, nobody has done that, and we are the first one to do such studies in Gold. Some more studies are pipelines. Once it is done, we'll put in the GEO site. So you have to wait for another 2-3 months.
Okay. And then we have one last question that I want to ask. What kind of opportunities do you see for data scientists and bioinformatics in this area?
What kind of opportunities? Sorry, what kind of opportunities?
What kind of opportunities do you see for data scientists and bioinformaticians in this area of male fertility research?
Okay, I think we have next panel discussion on that, but yes, it is a very good question. This the data scientist and the involvement on the infertility management or the oocytes, embryos is completely, it's a blank field. Not many people are working on that. Very few people are working, and it's an area which is worth exploring. And you look at this lecture, all the data scientists who are working on this are just working on some other field. The computational biologists are not working on this. Bioinformaticians are using the data and doing it. Not many people are working, and it's full of opportunity. There is a need for computational biologists to write code for specifically for this area, fully open. And one area which is a research area nobody has explored is working on the mechanism of how the oocyte activate, or it's completely, oocyte part is completely left.
Thank you so much, sir. I'm curious to know more about this. So for that, we would like to move on to the next part of today's symposium, which is a panel discussion. So to start with the session, I would like to invite again Dr. Andrew Lin, Professor, School of Computational and Integrative Sciences, Jawaharlal Nehru University, Dr. Mukesh K. Gupta, Professor, Department of Biotechnology and Medical Engineering, NIT, Dr. Pankaj Yadav, Assistant Professor, Department of Bioscience and Bioengineering, IIT Jodhpur, and the moderator of this panel discussion is going to be Dr. Mohit Mazumdar, Global Business Development Head for Pine Biotech. So over to you.
Thank you, Sri Gauri, and thank you, Professor Lin and Professor Professor Yadav Gupta, and thank you for all of you who have joined this panel discussion. So let me quickly just give a perspective about what we were hoping to discuss in this session, and then maybe we can start with having these different type of questions. So I think what we have been observing is that biological data is turning out to be much more complex than we ever thought of, and learning about it and correctly integrating these highly interdisciplinary subjects is quite a challenge. So with substantial and growing influence in health, environmental sciences and society, and it's being utilized by scientists from many diverse academic background. And that's what we have learned from Dr. Mukesh Gupta, Professor Mukesh Gupta, that there are so many areas that people are yet to explore because of the lack of understanding. So education in these areas is therefore like necessitates for effective development of skills in having interdisciplinary collaborations, communications, ethics, critical analysis of research. So all of these aspects, along with practical and technical skills. So I think that's where we wanted to have a discussion around that, the need for such training in education, and how are the ways we think is it possible to integrate some of these things, and at the same time what is the current scenario, because I think Professor Len is associated with Jawaharlal Nehru University, and Dr. Yadav here is associated with IIT Jodhpur, IITs, and Dr. Mukesh here is Professor Mukesh here is from a research institute, NIT, very famous research institute. So I think all of you have a various like different kind of experience having to work with different types of administration, and so that's how I think, if you can give us your perspective about first the use of data science and bioinformatics or biological data analysis for this newer generation who are started their journey in healthcare, life science students. So with that, let me, yes, I was just going to ask you, sir, that if you can just give your perspective.
Okay. So very briefly, I think that we should slot into 3 levels. The first is bioinformatics technical skills. The other is bioinformatics engineering skills. And the third is to be a bioinformatics scientist. Now, all 3 of them have very different inputs. Now, biology itself is very data-driven. But the problem is that the bioinformatician very rarely controls the research project. You have to come in as a scientist with control over the project, and very often you come in as a biologist, and then you have to get a bioinformatician or develop the bioinformatics skills as part of it. Now, the engineering field is now dominated by computer scientists. There's no way that a biologist or a life scientist can now compete with the algorithm development skills that come from computer science. So what most of us have trained in as students are people with enough technical skills. You've got a very good panel of skills that you have over there, and those would very easily be absorbed in biology laboratories because they have to handle the data that is generated by the laboratory or by samples that are sent out for omics-based, right? So that's, again, to summarize, 3 levels. Slot yourself into one of them based on your background and the skills that you can develop in the short term and long term. Technical skills, which you should be able to get with short-term diploma. The engineering skills, you need to have a background with computer science, and that does not mean just handling data. That means that you need to have algorithm development skills, which require far deeper understanding of the mathematics behind computer science. And the bioinformatics scientist, which is really, if you have a life sciences background, you understand what the data is going to mean, and you can develop and get hypotheses and also generate samples which can be used for generating the data. Okay, thank you.
Yeah, I think that's excellently put. I mean, categorizing them. Also, we get an understanding, I think, from this that some of the undergraduate students, I think, reached out to us about what kind of job opportunities that they will get after finishing such a degree and henceforth. So I think understanding that a research-based career would take a little bit longer time to develop. It's just not about the technology, but also to learn to apply that and do maybe a master's in research or PhD would be something that would be required for such student who's going through the research journey. And for technical skills, I think you would be needing an engineering degree or MTech degree, I think. Is that correct?
No, I was actually dividing technical skills and separating them from engineering. Technical you can get with a diploma. And I think short-term courses, you should be able to skill yourself with them. A few months, you can even do them online. And engineering would be that you have to have a basic skill set with developing algorithms. Very rarely can you do that in the isolation of your home. You need to have proper training with them. And you need to have a lot of online outside of university spaces, such as open source software development communities, Python groups that, for instance, Delhi has a very good software developed through hackathons. So, but it's a different platform from what we are normally used to. And the one that I said as a scientist is a slightly longer timeline. There's a lag phase where you have to do a PhD, where you incubate in institutions on big projects and you get the understanding. You normally would be in your late 20s or early 30s before you reach that stage. And there's an incubation stage through a PhD program.
Yes,
But I
absolutely.
would
Thank
say,
you. Thank you so much.
yeah, so just to repeat, if you're young and in the undergraduate, do technical skills. You can get them through online and you'll have to choose what you're going to.
That's a great suggestion, and that's something that also aligns with what we have been doing and asking students and researchers to be able to go through. So thank you, Professor Len, and maybe I now ask Professor Pankaj,
Yeah,
Dr. Pankaj Yadav, to
yeah,
maybe comment.
yeah. Thank you for inviting. Let me first thank Dr. Mohit for inviting to the panel discussion. I think it's wonderful to see Professor Andrew for after few many years. Okay, and yeah, I think I totally agree with Professor Andrew. So he has already given very clear answer to like what kind of skills to be is required. Yeah, sorry, I just was. Yeah, so I have just one thing to add, like what we now in IIT, what we are seeing and also we are implementing is like we are trying to make the things multidisciplinary. So those classical, the traditional way the people were defining the engineers and also those which are from biology background. So these boundaries are sort of now disappearing, and one need to have an understanding of different domains, not just a very particular domain in which you are trained, because nowadays engineers can also not work in isolation, so they have to have understanding of at least some fundamentals or some to some extent the knowledge of biology. And indeed, like nowadays what is happening is that nobody is working in isolation anymore. So you have to reach out to people who are expert in their domain, but at the same time you have to have some exposure and to have to extend your skills and knowledge to other domains also in order to excel, right? So I think in the coming generation, I think that change we will definitely see. And in as I said, in IITs now there is a huge focus on multidisciplinary trainings. So I think in this next 5 to 10 years, we are going to see a huge change in the skills with which people will be moving out in the job industry, right? So I think, yeah, that's my insight on this.
Yes, thank you, Pankaj, for sharing. I think that's completely, it makes sense. And in today's, even in job opportunities that we see looking at like diverse types of skill sets, as well as some work experience that is required by some of these industry or academic labs that are hoping that students are coming with some specific type of skill sets where before they apply and what they are applying for. So the question is about the integration in the curriculum. So what are your thoughts about like integration of, like, how do we integrate them in their curriculum? Or is it like for an undergraduate, how would that curriculum would look like? And for a postgraduate, how would that curriculum look like?
Yeah. Sorry, Mohit. Can you just repeat your query? Sorry, I just missed your voice.
No problem, no problem. So we understood that the way things are going and what are the different ways student can perceive this, and then we understand that okay, there are skill sets, interdisciplinary skill sets, and there is a choice-based credit system which is mostly have been adopted by many universities and institutions, giving a lot of flexibility to students to do interdisciplinary stuff and learn about different skill sets. I mean, overall, it's a good thing. So I'm asking that about the integration of such skill set based courses. So are we there yet, or what is the situation? So I know about IIT, but what are your thoughts about the IIT and other institutions that you might be knowing of? And like for undergraduates, how it looks like, and for postgraduates, how does that looks like now?
Yeah, so currently at undergraduate level, we have 2 years which are common for the all the branches. So all the students, they take common 2 years common courses, and after starting from third year onwards, they take some mandatory courses in their streams. And apart from that, there is a huge flexibility to opt from other domains as well. So for example, computer science student, a lot of computer science students, they take courses from bioengineering domain, right? So from third year onwards. And another thing that what we have at IIT system is also we offer from since second year onwards, we offer design credit projects and some kind of other projects that allow them to work in a multidisciplinary kind of team and work under the supervision of different expertise, so that they learn and grab that different domains by the time they pass out, right? So this is the scenario currently at the undergraduate. And for postgraduate, we also have the kind of since postgraduate it is more specialized, so it is expected they have some basic training already at undergraduate level. We usually have a few first year that is kind of coursework, and then followed by mainly the research projects. So that is currently the scenarios. But I think in coming years, once this NEP is kind of new education policy is implemented, there has to be some changements that will be implemented, because there are drastic changes that has been done. And to keep that in line with that new education policy, we have at IIT we have started with the lot of certificate oriented and diploma oriented training, so that the future generation is kind of trained on those high demand areas, and there will be a huge jobs market also once they take up those certificate and diploma courses. Yeah, so yeah, that is the thing at this moment.
All right. Okay. Thank you so much for sharing that. That does like makes us understand a lot about the way it is being perceived at a university or an institution level, and what are the different things that have been done and what people the students can expect from there. So I think that's about the students, but I have a follow-up question for Professor Gupta about the PhD students. I mean, for them, how are they? I mean, some of them are being biologists, and then there is a need for them to understand the aspect of data science and maybe exploratory analysis that they have to do for a hypothesis-driven research questions. So how you can train them, or what is the way we should go look at it right now for those students who does not come from that mindset, right? As you can understand, because you're going through and training those students a lot.
Very correct question. Now, when the PhD students, when they start, they have to come through the bachelor's and master's degree. And if you look at the current scenario, I'll talk about the India first. I've been looking after the admission of undergraduate as well as postgraduate in IITs, NITs, and all the JOSA and CCMT have been part of that. So as Dr. Andrew mentioned, we can distinguish two parts. One is bioinformatics, where the people are using the existing tools. They don't involve in developing a tool, rather using the existing tools. That's one set of scientists where we get most biological people comes in. People with the biology background, say BSc or MSc in biotechnology, or BSc, MSc in life science, or some other related branches. When they come in, then there's not many tools available, open source tools available, easy to use it, very user friendly. They're very nice user interface, graphical user interface. People can use it. And tutorials are very good. So all they have to understand is how to use those tools. That's not very difficult. People like us who are the initially, wet lab experiment scientists. So in order to migrate them as a bioinformatician, there are not many tools available. But certain tools which are not available in a, say, Windows-based system, that doesn't work well. Say, for example, if you're doing MD simulations and all, so it doesn't work so well in the Windows environment when they have to switch to Unix environment. So the students with the pure biological background find it difficult there. So that's the place where they're stuck in. So in those cases, the only opportunity, as others have also mentioned, they can take certain diploma courses available. There are certificate programs. Number of programs are available in IITs as well as the online NPTEL. They can utilize NPTEL training programs available. Many internships are available. So they have to do that. That's one way. But if you are going to write command-based tools for analysis, then there's no way, not easy job for the biologist. That normally starts with engineering. So if you look at now, many of the IITs and IIITs, NITs, they have started BTech program in data science and analysis. But BA programs are largely on, this is a high demanding program. Data science is very high demanding, but the expectation of the student is to go in AI and ML for various applications and then go to the other areas, non-biological areas. Some institutes like, say, MIT Bhopal or IIIT Allahabad, they do have courses specifically on bioinformatics. Those cases they took for undergraduate or the postgraduate level, where they need to have basic understanding of the programming languages to start with. So once you have that background in programming languages and understanding the basics of the AI ML, having background in basic, and then if you come to biology, the problem here is the other way around. These people are usually poor in biology. They will be very good at writing commands, very good at driving writing the codes for them, but what biological things they have to analyze becomes very difficult for them. And then there they have to go other way around. They have to look for the other biotechnology institute where they can be taught biology. Say I get many students coming and trying to understand neural network. So they come to us to say, understand about the neuron biology. That way it works. So there is a both give and take. Both has distinct pathway. So if you're going with the bioinformatics, there is a one pathway. Going with the computational biology, different pathway. But there is a scope to work together. See, I am basically a wet lab, and I have read books of the Andrew, and I have learned from that. So I am a student. Dr. Andrew probably never met him. He doesn't know, but I am an indirect student of him. I have read the books I have learned. So the learning is a continuous phenomenon, and PhD, as you know, is a tough thing. So if you love your job in a PhD, you can get things done. But you have to have a beginning, whether you are going to be bioinformatician or computational biology. Both requires help. That's how it will work.
Yeah, thank you so much. So yeah, I totally agree with you that Professor, it's a pleasure to have Professor Lin here with us, because his name in bioinformatics we know from the very beginning. Like from 2000, we have been hearing his name, and for a renowned persona in bioinformatics. So I think that it's a good question for you, sir, to tell us about opportunities in this area, like how you have seen this field evolving in a good way for people or students or researchers to take more participation in this, and what are the opportunities that are there in India right now as you see this entire field evolving?
That's not a very easy question to answer, just as the impact of the NEP is also not very easy because it's too disruptive a change. The big shift in India at the moment is the pressure and push towards entrepreneurship and startups. Even funding is in that direction, both from government as well as others. So I think that you should look at basically the panel over here. If I'm a teacher to many of the respected professors like Professor Gupta, I'm also too old to make such a comment on the future. But both of them have very good backgrounds, which I'd like to actually use as a base. See, Professor Gupta is a veterinary scientist of some background and has used data-driven science in his work over the last 10 years. And Pankaj is a much younger bioinformatician who's actually done bioinformatics as a PhD before coming back to the country to lead these kind of programs. See, the thing is that this was something of the past. What has happened with the large amount of data is that, really speaking, we can think in terms of some algorithms, but we really need to look at computer scientists to develop them. We can be, as Professor Gupta said, you can use tools relatively easy because we own the data and we're generating the data. Okay, so the answer to the question is, if I was 20 years old, what would I do? It's not that the excitement is not there. The research problems are really, really very exciting. For instance, the one that I started when I was 20 was on protein folding, which got solved with AlphaFold. And that was deep learning, right? So now these kind of research problems are being solved with many of the tools that we've come up with in computational biology. And because they are data-driven, there are models that we can use. Now, what is the kind of job that being enmeshed in these research problems as part of your training is a real opportunity for someone in the 20s. You can do it either as a PhD, but then that would be a longer term. So where are the startups? It's hard to say. I think they are largely in health diagnostics and in agriculture. So health diagnostics and not yet in therapy, because it's much, much easier to look for diagnostics, and AI is very, very big. Examples, for instance, can you use volatile matter? Can you use sound waves for detecting tuberculosis? What has already been done is image analysis, X-ray based identification for diseases using image analysis, et cetera, et cetera. So that's the low-hanging fruit. And a lot of startups are coming up in that area. I think ICMR has a panel of these kind of tools. And app development is another side field, because the actual push is what we call edge devices, which you have to have on the mobile. For instance, if you want a farmer in the field to detect what disease is present in the crop, he should be able to take a photograph from his mobile phone. And do you have the backend and do you have the tools to be able to detect that and develop that kind of tool? So startup space is the area that we really need to push towards. And if you're part of a larger program, the short answer to your NEP is that the 40% of your program theoretically can be done using online courses or courses outside of your main institution, and you have an academic bank of credits. The quality control is not very sure about what's happening over there. So really speaking, if you look at real quality, we're looking at what are you getting out of it? Have you been able to write out publications, for instance, in a research field? Or are you developing an app that is going into use? So if I'm taking two examples, let's say diagnosis. Can I detect disease from a cough sound from the mobile phone? Now that is already available as an app. Now it has to be rapidly developed as an app, and then it has to be scaled up. Now this kind of startup space is in incubators. I'm not sure of the quality of incubators within Indian institutions, but I would think that the IITs and the NITs are much better than a normal university, not in being able to scale up, not necessarily in the ideas or getting manpower. Okay, so that is where you should look. The short answer is, if I'm in my 20s, I would take a big risk and do startup space. I should get a salary in startup, and I should be able to learn on the side from platforms such as yours. And for that purpose, you should probably link up with institutions of repute like the IITs and NITs, so that some sort of the affiliation tag can be associated with the learning experience. And as Professor Gupta said, they themselves share a lot of their learning tools because NPTEL has been there from the early 2000s. The platform has changed because the internet is now 4G has made it far more permissible. It used to be based on desktop-based internet learning, which was not very easy. And but because of COVID, online learning has become the new normal, and we have to learn how to break with it. So back again, startup space.
Thank you. That was very okay. Sir,
Yeah,
you can go ahead. Yeah, please.
Yeah, I would like to add further to what Dr. Andrew said, that when we think of bioinformatics or when we think of computational biology, the general perception people take it just analyzing the DNA data or RNA data or protein data. That's that. But we have to move beyond that. Bioinformatics goes beyond that, including medical informatics, image analysis, sound analysis. Say, for example, in our country, it's not that popular. It's slowly picking up. But in other countries, say, for example, following the progression of cancer therapy. So a set of kits are available where, say, in one go, 384 genes can be tested for SNPs or whether the drug is working or not working with the drug. Similarly, for HIV, those tests are available where you need the analysis of the data sets to follow up whether this is working or not working. So one of my students was working on the Tata Medical Center. They use the bioinformatics for deciding whether the bone marrow transplantation has to be done in the patient or not to be done. That was to my surprise. What they do is they have the position called bioinformatician for that purpose. What they do is before transplantation, they go for the whole genome sequencing of that, and there are some marker mutations are there, which are suggestive whether the transplantation will be a successful or will be a failure. So those are known, and they try to analyze those mutations within them and then take a call. The whole panel will sit down there along with the bioinformaticians and the doctors and then decide whether the transplantation has to be done or not. So bioinformatics doesn't remain with the research level. It has gone to the medical field. I also recall now one of the protein powder selling company, that Muscle Blaze. No. So we had discussions with them. See, they are now coming up with the technology. It's there with others. So when you have to just take a saliva sample, send it to them, and they do a profiling. And there are a set of genes which they will analyze for specific for muscle tissue, specific for diabetic people, specific for lactose intolerance and so on, or the specific some predispositions to some diseases. Those kits are there, and then they do analysis of set of genes which may have impact on them, and then they suggest which product might be useful for you or what kind of diet will be useful. This is there in the market. That's the place where the bioinformatics is a must. Bioinformation is a must to analyze those genetic data. So it's there in the diagnostic, it is there in the healthcare management, it is there decided for the transplantation. And as Dr. Andrew said, sound-based assessment. The COVID time, IIT Bangalore came up with that idea. In veterinary field, if you go, then there is a livestock precision farming. It's all based on the data analysis. So when we think of the bioinformatics, I think we have to go beyond simply instead of thinking that the analysis and predicting a data set or predicting the molecular structure or predicting how the genes will interact. It has application and they are there in the field, less known to be.
Yes, absolutely agree to all of this. And this is like a really valuable suggestions and I think the audiences here are really appreciate the same, and I've been learning with all of these suggestions that you have shared with us. So I think what has been suggested is the understanding of the translational application, and now we see bioinformatics being used for product development. So I think when you talk about startup, it's about understanding that there is a need which is directly related to the people, and you can think about these different problems and then think about a product that could help them with their problems, and that product is coming from data analysis. By basically bioinformatics, from the very beginning, what we were doing is like doing a lot of comparison. So that helps us understand how it could be useful for a variety of like personalized problems that we are trying to figure out, from nutrition to what kind of food we should eat, whether I would get, whether I can lose weight or not. So there are so many areas where there's a whole lot of business that has been done through supplements, through all of these different things that are there in the market, where there's a need of more specialized products with the help of data that gives us very great insights and is much more reliable and trustworthy to go for. So I think, yeah, and that's absolutely something that I think everyone should take a note of and start thinking in that direction. So thank you so much for those suggestion. So, and we
Mohit, I just want to add two more things. Like one thing that I think both panelists have already greatly emphasized. So this is what we are talking about: digitalization of healthcare. So digital healthcare, it is currently actually totally in very demand, and in coming years we will see that a lot of digitalization will be happening, where we can use apps and etc. to even check several do a lot of diagnosis at ourselves as well. So digitalization is a huge going to be a huge demand for the upcoming generation. Also in the bioinformatics will also, a lot of bioinformaticians will also be kind of being involved in this in this domain. And in India, I think what we are going to also see is that there is a huge kind of increase in the volume of the data that we are going to generate. And there also we will need a lot of people who should be skilled enough to handle and analyze that data. So I would just say that to the audience that there is a huge scope, and a lot of opportunities will be opening up in the coming time. So yeah, so I just want to motivate them.
No, thank you, Pankaj. I think we are in sync. I was going to ask you the same things, but you rightly summarized the entire panel discussion, and thank you so much for all the panelists for participating today and joining us in this research symposium. We are so much grateful, and I think the audience have really benefited. We have a lot of students here, and we are also live on our YouTube channel where there are a lot of students there as well. So thank you so much for your time, and thank you so much for participating and giving us your insights from your experience. So we would have another session, next session, which is a student poster presentation. I think students are really excited for this part of the symposium. And without further delay, I'd like to invite Sri Gauri to summarize the panel discussion and move to the poster presentation session. And again, I mean, I personally want to thank Professor Lin and Professor Mukesh and Pankaj for their time and for these wonderful discussions. Thank you.
Thank you. Thank you, Mohit. Thank you for having us.
Thank you.
Thank you so much, everyone. Dr. Lin, Dr. Gupta, Dr. Yadav, and Dr. Mazumdar. That was such an engaging and insightful panel discussion. I want to take this opportunity to quickly summarize the points that were discussed. Firstly, we discussed the need for data science education for all life sciences students and equipping them with tools and skills they need to analyze and interpret the vast amounts of data generated. And we also discussed about how it is impacting healthcare, biological, and life science education. And we also discussed the integration of hybrid learning and challenges faced by universities in implementing. Taken together, I want to thank our esteemed panelists for taking the time out of their hectic schedule and sharing their knowledge and expertise on these topics. So far we discussed about some of areas of academic research in biological data science. And we also covered some of the ways one can get involved in these research work. But students worldwide, they often lack the opportunity to showcase their research work after completing a project. Therefore, our symposium encourages students to participate in a poster presentation competition, which is a great way to share their research project, interact with fellow researchers, and get instant feedback on their work. It also serves as a valuable networking opportunity and allows them to forge new relationships for future collaborations. And this year's theme for poster presentation were Omics and Bioinformatics, AI, machine learning and data science, astrobiology and space omics, computational biology in biotechnology and pharmaceutical industry. The submission was open to all registered participants from high school, undergraduate, graduate, and postgraduate, as well as for citizen science projects. Now, let me also take a brief moment to introduce you to the panel of judges who will be reviewing the project works. Dr. Ramesh Kumar, Associate Professor, School of Allied Health Sciences from Reva University. Dr. Priyanka Narar, Assistant Professor, Institute of Biotechnology, Amity University. Dr. Julia Panav, Associate Director, Tauber Bioinformatics Research Center, University of Haifa. And Dr. Mohit Mazumdar, Director and Data Scientist at Pine Biotech USA. The presentations will be judged in 4 categories, that is introduction, methods and results, discussion, conclusion, and finally, the presentation quality. The best posters will be selected by the judges for prizes in different categories: best technical research project on a competitive topic, promising research direction for future potential, and finally, most innovative approach in an established domain. The guidelines for the competition are very simple. Participants will be given 5 minutes to present their poster. Once the time is up, we'll have to stop your presentation and move on to the next candidate. So kindly make sure that you complete your presentation within 5 minutes. So with that, let us invite the very first participant of the poster presentation competition, Ariana F. Rahman, who will be presenting her project on a novel deep learning predictor for post-therapeutic breast cancer recurrence risk using a multi-omics data integration framework. So over to you, Ariana.
Hello, my name is Ariana, and I will share my screen. Okay. And this is my poster. And my project is on a novel deep learning predictor for the post-therapeutic breast cancer reoccurrence risk using multi-omics data throughput. So, over the last few decades, many breast cancer patients have become disease-free because of the many advances in early detection, in diagnoses, as well as treatment of breast cancer. However, for this breast cancer can come back for almost 30% of patients who were disease-free after initial treatment. And this is called reoccurrence. So reoccurrence can return after post-treatment, and current methods of reoccurrence risk prediction use clinical or phenotypic characteristics, such as age, tumor stage, or menopause, and other biological factors. And while these characteristics are certainly helpful, they may not give a complete understanding of the underlying genetic causes for the reoccurrence. So by using multi-omics data, risk prediction of tumor reoccurrence can be used to make effective treatment decisions and aid in the survival outcome of patients. So ultimately, the aim of this study is to use this data in order to identify a marker list of gene variation and an expression for the patient stratification based on their tumor regression status. So in order to find these genetic causes or underlying, first we have to take a look at the data. And that's why I conducted PCA to understand the features of the data. And here we can see that the data clusters around the ER status, whether it's negative or positive, and it does not cluster around disease-free or reoccurred. So based on the observations above, the estrogen receptor status seems to be masking the data representation of the samples due to their tumor regression status. And so in order to correct for this confounding factor, an autoencoder, which is a deep learning non-linear based embedding approach, was used to get the single representation from both the omics data, like copy number alteration and the mRNA data, while also simultaneously adjusting for the confounding factors. And so this integrated data from Amy is then taken and applied to random forest, which will classify it into disease-free and tumor regressed categories. So once the data was corrected for the ER status, the accuracy of the model was actually at first, without adjusting for it, almost like a coin toss, approximately 50%. But after it was adjusted for, then that accuracy rose to almost 96%. And this was after cross-validation with multiple data sets outside of just the TCGA, the cBioportal dataset I used. And so what the ultimate output of this is the 25 most highly important genes toward the tumor reoccurrence, which is shown in this heat map above. And some preliminary conclusions can be made, such as some of these genes are more immune related, such as STAT1. And so this might tell us a little bit about how reoccurrence might be more about the body's ability to fight off the last remaining cells that are cancerous, rather than how initial treatment, which is using like radiotherapy or chemotherapy to fight off the initially cancerous cells. And we can see that in the PCA, after it has been, the ER status has been adjusted for, that it no longer separates, it no longer clusters by the ER status. And instead, it now does cluster in the disease-free versus the reoccurred. And eventually, the clinical application of this would be that this was made into a pipeline that takes the tumor recurred or the disease-free patients, their data, and through the autoencoder and with only 25 genes, it will give the probability of a patient from of how probable it is that their cancer will reoccur. And I use that in a web app. So this web app, with the information that the patient provides, will generate a probability score from based on their either mRNA data or their copy number alteration data. And this will definitely be helpful toward improving diagnoses accuracy, along with the initial predictions of the treatment plan, as well as helpful in clinical decision making. And in low resource settings, it can be used for a much more speedy reoccurrence prediction. And this doesn't just have to be limited toward breast cancer specifically. It can be applied to many other cancers as well. And it can also include other omics data. So my project initially used mutation data and microRNA data as well. But since the sample size was so limited and it was very sparse, they weren't covered in this project. But eventually, a more diverse integration strategy could combine other data sets that have mutation data and microRNA data in order to make a more multi-omics project. And it can go beyond the genetic level as well into the central dogma. Like it can go into a protein-to-protein interaction network that can be used for further therapeutic applications. And that's my project. Thank you for listening, and I'd be glad to listen to your feedback.
Thank you so much, Ariana. I would like to pass on the stage to Dr. Julia Fano to hear her feedback about the project.
Thank you very much for sharing. It was very interesting. I don't think I have a full understanding of I forgot the name, Amy, right? Of the exact procedure that you do that you do to differentiate. But can I ask you why exactly you wanted to take away this factor of ER? I mean, you saw that it differentiates on the PC, right?
The PC.
Right, but maybe it does have, a biological meaning that the sample. So why did you anyways decide to take that factor away? Just a question.
No, totally valid. And ER status is very important for breast cancer, specifically like women with their estrogen receptor positive. It's been shown like their cancer affinity, and there's of course linkages to this status. It's very important, but in my data, it was clustering by the ER status, and this was like masking the data representation because it wasn't clustering by these disease-free versus reoccurred. Sorry, this is kind of small, but there are like circles and triangles, and that's like disease. Oh, sorry, no, I mean, I made a separate chart. So here, here, like see how all the red and blue are like mixed together? And this is by like the disease-free status. So all of it's like mixed, and it doesn't show like separation between disease-free and reoccurred. So what's causing the reoccurrence or these genes, like ultimately the PCA shows features, which are the genes. And so if it doesn't cluster by disease-free or reoccurred, it's not showing which genes are the most important toward this reoccurrence. And instead it would cluster by the ER status. So definitely, like you said, the ER status is important, but since it's causing the distribution to not cluster by the disease-free and reoccurred, it's therefore it can't be seen what the top genes are in order to find what the reoccurred, like find the best genes for reoccurrence prediction.
Very impressive work, I must say. I'm really impressed. Thank you very much. Very nice.
Thank you so much, Dr. Panal and Ariana. So with that, now I'd like to pass on the stage to the next participant of the poster presentation, which is Umesh Kumar. He will be explaining this project on exploring the microbiome in cat and understanding the role of microbial communities in feline health and disease. So that the stage is all yours. You seem to be on mute. Could you please unmute yourself? Yes.
Hello, everyone. So the topic that I'm going to cover today is like exploring the microbial community in cats itself. So as you all know, it's like most of the diseases nowadays is actually spreading. It's kind of like a zoogenetic zone. So most of the times, like the bacteria are actually not just bacteria, but even viruses are actually transmitting from a different species to human being itself. So in order to understand the future, predict the future diseases that is going to occur, as you all know, like we are more close to felines, like including cats, dogs, and so most of the diseases that is going to be like spreading in the generation in the coming time will be also from cats itself, cats or dogs itself, like the most probable. So in order to identify such kind of potential candidate, like the mutations that is going to happen in the near future, like I am conducting a study which actually disclose the number of bacterial community that is there in the cat species itself. So I actually done this using TBioserver only. With a data to analyze tool, like I actually found close to I actually studied a genomic data, which is containing like 4 different samples. And that sample is of fecal samples or pharyngeal samples, brongovascular fluid samples, and also from blood samples. And the data is actually for 6 different kind of cats. And that also include males as well as females. So in all this study, like after going through the analysis, like I found that more than 70 to 80 bacteria are actually there in the fecal sample alone. So most of the times, like most of the like from my perspective, like I thought like the most of the samples will be there in the fecal, but actually the thing was not like that. Like it was actually from oropharyngeal samples that I found the most number of abundant communities out there. So more than 80 plus bacterial communities were there in the oropharyngeal samples, and fecal sample was the second highest, followed by bronchovascular fluid samples. And in most of the samples, the bacteriocides like was you can actually find bacteriocides in quite a lot number. Like when we actually discuss about the fecal samples, like the most of the abundant community was from Bacteriocidae family. In oropharyngeal sample, it was actually from a Pasteurella, and in bronchovascular samples, like the Pseudomonas was there, and in blood sample, Spingobacteria were also with there, like as in abundant. So even with that, like even you can from the analysis, like we actually find quite of like different kind of um, data itself, like the data that is the bacterial community that is actually there in the male samples was not seen in the female sample. If I would like if I would point it out, like some species of Bacteriocides was more abundant in female cats compared in fecal, but in the male sample it was more of like Bacteriocides sample. If you Fusobacteria sample was more. And when it comes to oropharyngeal sample, Pseudomonas was more in male compared to Pasteurella sample in female. Even the same trend you can actually see in bronchovascular sample also. Like Pseudomonas was more in female cats, while Spingobacteria were more in like blood. So with that in mind, like that actually helped me to just like come to a conclusion, like even with the genders, like the bacterial community can actually varies, even with gender, even with the physical condition, and even with the different sites itself. Like the fecal, as I already told, the fecal was the exception. The expectation was the fecal would be having more bacteria, bacterial community, but oropharyngeal was the most abundant. So this actually proves that, like as the oral region, like oropharyngeal or the oral region that the cat used to like lick or even bite us, like in the future generation, like more possibilities are actually there from that like from the feline, like more different kind of bacteria can even the possibility of transferring is there to humans are there. And in the future analysis, I would like to study more about the bacterial community, the different kind of gene that is actually there in the particular family, which can actually transmit it to the human community itself. And if from that analysis, I can say that, like by stopping the future, by studying the future mutation, like mutations and all, we can actually find a different kind of treatment planned in the coming generation itself. So that is all about my posture. If you have some query, like I would like to answer your queries itself. Thank you.
Thank you so much, Umesh, for that presentation. And to share judges' feedback, I would like to pass on the stage to Dr. Priyanka Narad. Ma'am, could you please share your feedback?
Oh, yes. Am I audible?
Yes, ma'am, you're perfectly audible.
Okay. Thank you, Umesh, for your presentation. And it is indeed a nice work. I just have a few questions for you. The first thing is that in your poster, you've taken one sample, right? So how
No,
many?
actually, I took
Yeah.
like 4 different kind of samples, including like 4 different kind of samples from 6 different kind of cat species. Cats
Cat
itself, six
species
different
coming
cats.
from a single study because the accession number is 1. So I believe you've
Yes,
taken from one study, right?
yeah, one study only.
Okay.
Actually, it
And
was actually having like a 6 different kind of cat species. They actually study like 6 different kind of cat species in that.
what was the total sample size? How many samples did they process? 6.
They actually no, they actually proceed like more than 80 plus. 80 plus samples were there.
Okay. And since you are talking about males and females, did you also try to look at how many males and how many females were in the sample
Yes,
size
like
in order to check?
total, yeah,
Yeah.
total 4 females were there and 2 males were there.
4 females and 2 males.
Yes,
Okay. And 80 samples, right?
yes,
Okay. And then you process them using?
T-Bio.
You process them using, sorry?
T-Bio server.
T-Bio info.
Yeah, yes.
Okay. Okay, great. So these were 16S RNA. Was there metadata also available? Did you look at that?
Yeah, metadata was also be available there.
So in your analysis, did you also include the metadata, or this is just based on the bacterial coabundance?
This is actually based on bacterial coabundance only.
Okay. So any of these bacteria which you have identified, any of this is something which is not known prior in cats? Did you get something which is
So there are more than in fecal sample, I actually find more than 50 plus that were not been discovered in any of the studies before, like 30 plus in oropharyngeal sample, then close to 10 in blood, and in 30 in or bronchovascular sample itself.
okay, great. So these are also gender specific.
Yeah, these are also gender specific.
Okay. So just a suggestion, you can go ahead and find out whether these bacteria are going to be beneficial or causing some.
Even I had the data, like I didn't add this in the poster because I was yet to publish this paper itself. So actually, like even the different activities that they actually like that is actually creating in the cat's body itself, I actually noticed few of them.
Few of the changes that these bacteria are causing.
Yes,
Yeah.
exactly.
Yeah. So positive or negative interaction can be explored.
So more than like if I would like to say in total, like more than 40% the bacteria were like beneficial, and 60% the
Good
bacteria were like 60% bad bacteria, and 40 more than 40% like good bacteria.
Okay. Okay. Thank you. Great work, Omish. Thank you.
Thank you.
Thank you so much, Dr. Narad and Umesh, for that wonderful post presentation. And next, I would like to invite Shimasha Indani, who will be presenting her poster on exploring the role of circular RNA and microRNA gene interactions in Parkinson's disease using transcriptomic profiling of peripheral blood mononuclear cells. Over to you, Shimasha. It looks like you're on mute. Could you please unmute yourself? Could you please unmute yourself? I have requested us to unmute on the Zoom. We are still not able to hear you. Chumasha, can you hear us? All right, perhaps while you're fixing your audio connection, we can move on to the next participant. So next participant is Tanmoy Biswal, who will be presenting his poster on exploration of the intestinal microbiota regulated by high fiber and high fermented diet. So over to you, Tanmoy.
Yeah, thank you for providing me with this opportunity. I'm just getting ready my poster.
Yes.
Yeah. Can you see my video?
If we can see your screen, yes.
So actually, now let's go to the introduction part. So this is about the factors that influence the gut microbes, the health issues that they cause, the technology that can be used to determine these gut microbes, the positive changes that they have on our life, the chronic and the diseases, the interactions with immune systems and immune responses that they play. There are several thousands of gut microbes that play a key role in immune system, developing our immune responses, training our body against foreign invaders. So the introduction part just gives us a brief information about that. So I have used the T-Bioinfo server's Time2 pipeline and R programs to develop this project. So first of all, you see in the top picture that is the functions that the gut microbes performs and what role that they perform in the body. And second picture is about the microbiome's effect and how it is processed and the primer setups that is typically used in while determining this kind of analysis. Now we come down to the metagenomic component analysis. Here I have used the Time2 pipelines, and the pipeline is totally being explained here from every step by step. So I'm not going into the details of the pipeline. So pipelines basically gives us OTUs, that is operational taxonomic units, and ASVs, that is used to determine the taxonomic proportionate abundance plot. Then we go to the 16S downstream analysis of rRNA data, mRNA data. So this gives us OTUs where we take out that OTU tables and ASB tables and compare it with the metadata, and then we get to know about their diversity, their abundance from the metadata plot. Then we can do statistical analysis using statistical analysis of that taxonomic abundance bar plot. So this is the downstream analysis. First, we do the downstream analysis. We have to load the libraries and the metrics. Then we unify the informations that are present in the OTUs and the ASB tables. And then we get the taxonomic compositions of the samples using alpha diversity, beta diversity, and we got the taxonomic abundance bar plot. Now, as we come down to the results parts, the results are clearly interesting to me. I did not expect this kind of results. The alpha diversity is basically measures the diversity within a single sample. So I did not see any change during both the diets. Both the diets did not show any significant change using Shannon and Chow diversity, but in while measuring the time points, there was some changes in the time points, and the people consuming the fermented diet shows much more microbial abundance compared to the fiber diet. Now, when we come to a beta diversity was showing changes between the first week samples and the 9th week samples, between the high fiber and high fermented diet. So these studies were not clear what were the exact role. Then we dig down to the phylogenetic tree analysis, the phylogenetic tree analysis. For that, we have done the phylogenetic tree, and it suggests that some phylums of some phyla of Bacteroidea, class Bacteroidea, showed more growth in fiber-rich diet, and one was showing in fermented diet. So from there, we can draw a conclusion that lower groups, such as the order, class, or family levels, must have shown different kind of bacterial diversity there. So I have gone with the proportionate taxonomic proportionate abundance bar plot in this comparing the major phylas. And there was an important thing to notice was that out of 6 samples in the 9th week, 3 samples of high fiber diet were completely missing actinobacteria, so which was not overall good in the perspective of the studies. So when we come down to the statistical analysis and finding the unclassified genuses, species, and families of these, so there were about many in the family level. We are getting some little bit of the median fiber was that 2.4, and the median of fermented diet was 4.5. So fermented diet was having much more unclassified families, and same with the genus level. And when we come down to the species level, it was just out of 1,255 species were present in the OTUs. 60 to 70 were marked unclassified. In some samples, it was down to 48%, and some cases it was up to 90%. So this is the data chart that we present that we get from that. Firmicutes were less abundant in high fiber compared to fermented, and actinobacteria, which is showing much more in fiber diet, but at the later stages, the actinobacteria just disappears from 3 of the samples. And when we come down to the discussion and conclusion part, is that there is less abundance of unclassified families and genuses of bacteria and fiber diet compared to fermented diet. And we also know that proteobacteria that plays a key role in gut health. So proteobacteria level was okay, but the actinobacteria was missing in 3 samples out of the 6 in the final week. So, and further thing that it is very much hard to understand from this study that what is the exact role of gut microbes, because we don't know species level interaction of almost every sample, because 60 to 70% of the species are still missing, and we have to discover it using the wet lab techniques. And without understanding their role, we can't get the role of, we can't get to know the role of the true potential of the gut microbes that is present in our body, and what kind of exact functions that they do, and how they interact with our body in training our immune systems, in developing other functions, that is synthesizing of vitamins and production of small chain fatty acids, etc. So we have to get to know more about these things. And I would be, this is just the tip of the iceberg, as I'm saying, because much more thing has to be discovered.
Have less than 6 minutes to finish with your course. Okay.
Okay, ma'am, I'm done. So it is very much important to understand the role of the gut microbes, and further studies using single cell RNA, single cells RNA status that we can discover more about this. Thank you, and I'm ending this, and I'm listening for your feedback that you can provide me.
Thank you so much, Tanmoy. So to attend to your poster, I would like to invite Dr. Ramesh to share his feedback for Tanmoy's poster.
Yeah. Okay. Okay. Is it audible? Is it audible?
Yes, sir. You're perfectly
Yes,
audible.
Oh, wow. Okay. Okay. So Tanmay, okay, you have done very nice data analysis, but I have one query over there. So you did that fiber diet that and then fermented diet analysis, is correct? So this fermented diet or fiber diet, which were actually inducing, it's like increasing that alpha diversity more, or it is a beta diversity more. So because that is actually that good bacteria. Okay, so it's important for which we have to prepare like a fiber diet also, and even fermented diet also. So what is your opinion to increase that alpha diversity? You have to take that more fiber. Suppose you want to increase that beta diversity. Suppose like a good bacteria it is belonging to beta diversity. What is your opinion?
in my opinion, is that we should take a balanced diet, because the groups of bacteria in each have their own functions, and they provide their own ways of, if there is an imbalance, so the body functioning may be hampered, because both groups of bacteria, whether it's fiber or fermented, they have their own roles, and they should not be changed. Means you can do it for a experiment point of view, but they play a crucial role in human health. So I think people should have a balanced diet instead of going for high fiber or high fermented. So balanced diet is required.
Okay, okay, okay. It was very good analysis you have done, but yeah. So that is fine, Carry. Otherwise, all are fine. We have done that. Okay. Okay. Thank you. Thank you so much.
Thank you so much, Dr. Ramesh Kumar and Tanmoy, for joining the post-presentation competition. So with that, let me quickly share my screen and start the next session. All right, so I hope so far all the attendees enjoyed the first part of Omics Research Symposium 2023, which focused on bioinformatics education at Omics Logic Asia. We have learned about the various platforms and initiatives that are shaping the future of biological data analysis. Now it is my great pleasure to invite you to the next part of the symposium, which will focus on Omics Logic Africa. The session will showcase the various initiatives, challenges, and opportunities in the field of biological data analysis in Africa. So without further delay, please join me in welcoming our next speaker, Mr. Adekunle Adeluboye. Mr. Adeluboye is a molecular scientist and academic who is equally a tech enthusiast with a professional background at a medical laboratory. I would like to request the participant to please mute yourself. Thank you. Currently, he is a faculty at the Lead City University, Nigeria. He is also the founder of the Research Mentor Program, Undergraduate Research Education and Knowledge Acquisition Traineeship. His ongoing research interest includes molecular biology and genomics, with a focus on molecular characterization of hepatitis B viral infection. He is also presently the director of Omics Logic Africa program of Fine Biotech, USA, a project that aims to provide ease of access and tools for capacity building in big data science for teaching and research across Africa. So with that brief introduction, now I'd like to pass on the stage to Omics Logic Africa. The stage is all yours.
Hello, thank you very much. I'd like to please ask for permission to share my screen.
Sorry, you already have the permission to share your screen. Can you please check?
Okay, thank you. Please let me know when you can see my
It is loading up.
Yeah, we can see a screen, but it's not proportional. So.
Yes, now we can see your screen.
Is it better now?
Yes.
Okay, great. Good afternoon, everyone from this part of the world. I'm happy to be part of you this afternoon to participate at the Omics Research Symposium, the 2023 March edition. And in the next few minutes, or let me say an hour or so, you will be with us here on the Omics Logic African team, where we'll be sharing some of our works and equally have notable speakers from this part of the globe speaking to participants across the world. So once again, I want to welcome all the participants and to this Omics Research Symposium. And I want to appreciate the Omics Logic Asia team for what they've been doing since the beginning of this program. So quickly, we are moving on into the African session within the next hour or so, and we'll be looking at the Omics Logic Africa programs, scope of the bioinformatics programs we are running here, as well as researches within the African continent. We will equally be taking two important keynote presentations by two notable scholars of repute. The first person will be Ashraf Akintola, who is a fellow, and he will be talking to us on the need for more bioinformaticians in Africa. Also, moving on, we'll be having Dr. Oluke Ojo speaking with us on the role of bioinformatics in livestock improvement here in Africa, particularly from her own research endeavors. Dr. Shola Ojo doubles as an academic and also an industry professional, being a registered animal scientist. And so she'll be sharing with us from her works, and we hope to have a good time, with that session. And last but not the least, under the Omics Logic African session, we will be having a panel session where we'll be having some, panelists discussing around capitalizing the potential of bioinformatics research for Africa's economic development. But before then, quickly allow me to introduce myself. My name is Adekunle Adeluoye. I'm a faculty and lead, with the Leeds City University here in Nigeria. I'm equally an associate of the Medical Laboratory Science Council of Nigeria, as earlier introduced. Currently the program director for Omics Logic Africa. And so moving on this afternoon, I will be talking to us about the Omics Logic Africa programs within the next 10 minutes, where I'll be giving an overview introduction on what we've been doing on our training programs, and also talk about partnership strides we had with several institutions to advance bioinformatics on the continent. So the Omics Logic set of bioinformatics and big data analysis training programs of the Biotech USA, which has been globally proven by peer reviews and curriculum experts in partner institutions that the program has been conducted, explores to help close the need gaps that exist with regards to bioinformatics education and training, globally. And of course, this is of great importance for us here in Africa in terms of, building these scientific capacities among students and young professionals, as well as researchers and faculties across the continent. And so the postulation we are working with at Omics Logic Africa, that is anchored by Eureka, is that by increasingly advancing the knowledge of bioinformatics and its use in obtaining scientific insights from sequence data, there would be a significant growth and development of the bioinformatics community in Africa, and also its considerable implications for translational research and practices, that will help improve healthcare and other key, functional areas, in the scientific community and the society at large. Of course, we're looking at, how this can impact on biology generally, agriculture, medicine, and several other aspects of human endeavors. And so the goal of Omics Logic programs is therefore to provide this training in bioinformatics to enable our students, clinicians, as well as faculty and researchers of all backgrounds to develop these novel independent research guided, skill sets, and, be able to utilize these to, advance their work and practices. And so the Omics Logic program boasts of everything a participant actually needs to get started and grow in the bioinformatics and big data analysis space for scientific research and practice. Our program, of course, combines online interactive sessions as well as self-guided study materials, practical assignments for an immersive experience, all of which have been proven to be effective in our well-known and respected Omics Logic bioinformatics. Hello, can you hear me?
Yes, yes, we can hear you.
So why the growing? Hello, can you hear me? Hello, can you hear me?
Yes, 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 we can hear you adequately.
Yes, we follow.
Okay, sorry. I think there was an internet glitch there. So, why the growing interest in bioinformatics and big data analysis continues to grow in the African region? Definitely, we at Omics Logic Africa and Eureka, we are positioned to provide the plug necessary to help many participants and researchers and students gain this, skill set for utilization in their own personal research and scientific, endeavors. And the focus of this, of course, is to help catalyze development of bioinformatics researches and omics science generally within the continent, and also to subsequently, of course, benefit the entire continent with the output of these scientific endeavors. And so we are glad that we are the Omics Logic African team with our training programs, anchored by the Pine Biotech Incorporated USA is, helping to fill these gaps that is, found to exist, particularly within the continent from the perspective of infrastructure, gaps. And some of the programs we've been able to, coordinate within the space of our practice over the years include data science for biologists as far back as, May 2021. We also organized data science for biologists program, in 2022 as well. And equally, we've had several introductory programs, introduction to bioinformatics training, where we bring, participants from different backgrounds to come to appreciate the import of bioinformatics, in the science environment today. And lately, in December 2022, our latest program was the introductory bioinformatics workshop, which has about 392 participants that range across, several countries here in Africa. And we equally have some participants joining us from, other parts of the world, including Pakistan, India, and some other locations around the globe. It was a huge success. A program that was designed to introduce many to the field of bioinformatics. And again, we organized, other high impact trainings such as Omics Logic Genomics Workshop, as well as Omics Logic Metagenomics Workshop, all of which, had participants moving on from these basic trainings to our research fellowship program. And so some of the training program participants that has gone through the program includes Theo Matthew, and these are some of the laurels that these participants have gone on to accomplish while undergoing our training. We equally have Ujubi Sola, who is a pharmaceutical microbiologist, and I've seen her practice and application of key bioinformatics concepts in her practice, and significantly enhanced. All right, including we also have Gisele Flodere, who is from Cameroon. Okay, so like I said, we cover, we organize these trainings and bring a lot of participants across the continent to participate in our trainings. So Gisele Flodere, have seen herself prepared to take on her research in her, postgraduate PhD training program. And of course, the Omics Logic training played some significant roles in that endeavor. So we have also been able to forge notable partnerships with institutions towards integrating our hands-on mentor-driven and globally proven training methods and resources into university education, both at undergraduate and postgraduate levels, so as to help scale the growth of bioinformatics and the use of bioinformatics in researches across these institutions. I'm glad to say that we also have some other institutions, in the works in the recent times. We are having talks with them, among which are University of Port Harcourt, as well as University of Ilorin, and also Lagos State University. Suffice to mention that similarly, Omics Logic African team has participated in a number of scientific workshops and trainings, conferences, one of which is the Nigerian Bioinformatics and Genomics Network workshop, where we presented on experimenting with 21st century e-learning design for bridging bioinformatics education gaps in Africa, using the Omics Logic as a test case. And moving on to, I'd like to, at this point, mention our next training program, which is around the corner. It is titled Bioinformatics Workshop from Training to Project Design and Publishing. This is a program that seeks to start from the introductory beginner level with participants in that workshop, and ultimately, move the participants to the focused trainings with different specialization that applies to individuals. Talk about metagenomics, genomics, infectious diseases, predictive medicine, okay? And ultimately lead these participants to the stage of designing their own project and moving on to presenting or publishing their works. So that training will be starting next month. All right. And so I'll conclude by saying that Africa is considered to be the most genetically diverse continent on the planet, with the potential to contribute significantly to the field of genomics research. And I'm happy that we are beginning to, see more and more interest in this respect on the continent. And that is notwithstanding some of the challenges that we have on the, with bioinformatics on the continent due to limited resources and infrastructure. However, there's a greater need for investment and collaborations in order for us to fully harness Africa's potential in the genomics research space. And I'm happy that the Omics Logic team is actively playing such an important role here, because bioinformatics has become an indispensable tool for management of these hugely generated data sets in our laboratories. Moving on, I look forward to more and more collaborations with institutions and individuals, as well as participants willing to conduct their research, with Omics Logic African team. I'd like to, at this point, acknowledge the Omics Logic Africa and the Eureka team. These are a team of individuals working hard to advance the Omics Logic training across Africa and also benefit some other countries that come to join our programs. So on this note, I'd like to say thank you for listening into what we've been doing with the Omics Logic African programs. So at this point, I think I want to hand over to our community engagement manager, Ayonfeolu Alabetutu, who will be coordinating the next set of speaking engagements we have with notable speakers we want to learn from this afternoon. Over to you, Dr. Ayonfeolu. Hello, Dr. Ayonfeolu, can you hear me?
All right, thank you so much. I can hear you clearly. So I will be sharing my screen now in a bit. Thank you so much, everyone, for joining us. I would love us to, at this point, get ready. I see that the next speaker is quite on ground. Wow, that's great. So I will be sharing my screen in a bit and we will get started. Okay. All right. I believe you can see my screen now, right, everybody?
Yes, we can. Yes.
Hello, can you see my screen? So once again, I welcome everyone, every participant to this Omics Omics Research Symposium with a thematic focus on bioinformatics and data science research and training. The African session focused more on the African ecosystem, and I trust you would actually enjoy what we have brought to you. I am Ayonfoluwa Alabetutu. I'm a molecular biologist and genetic expert from Penza State University with a pass for population genetics and so on. I will talk more about myself in a bit. Yeah.
Hello, are you there? Hello, can you hear me?
Yeah, we can.
Yeah, we can hear you.
Okay, I think he might be having some challenges there, so I'd like to take it up from here. So in our next session, we want to invite our first speaker to take the floor now. Allow me to quickly share my screen so I can take over from there before he comes back. Okay. So our first speaker for this afternoon is Ashraf Akintola. He will be speaking to us on the need for more bioinformaticians in Africa. And Ashraf Akintola is a researcher, an entrepreneur, author, poet, and public speaker who has led fellow colleagues to conduct free online training and courses in bioinformatics for beginners and middle-level mastery in bioinformatics skills. He is a PhD fellow with research interest in biomedical convergence, science, and technology, with a specialized focus on mitochondrial genomes of mosquito species of high medical importance. And Ashraf also leads an impact development company with the mandate of leading a generation of impact, prosperity, and sustainability among Africans in the area of bioinformatics, data science, health, education, media, and publicity. Please welcome with me Ashraf Akintola, who will be taking us on the next topic, the need for bioinformaticians in Africa. Hello, Ashraf, are you there? Can you hear me?
I have here.
Oh, great, great. Please, you have the floor. Take over from here.
Thank you so much. I really appreciate the session that I've been able to join so far. I would like to share my screen to start with. I think my screen is visible now.
Yeah, we can see it.
Thank you so much. Thank you so much, sir, for the introduction. I remain my humble self, Ashraf Akintola. I am a PhD fellow at Kyungpook National University here in Daegu, South Korea. And I have been given 30 minutes to discuss the topic, the need for more bioinformaticians in Africa. I first of all want to appreciate the Pine Biotechnology Company in charge of the Omics Research Symposium for this special gathering to bring about different researchers in different areas of bioinformatics, especially from Africa origin, of African origin. So it's important that once this kind of forum is rampant and consistent in our society, a lot of people will get to know the benefits of being involved, or what's the bioinformatics as a whole, what's the prospect it has for humanity and especially Africans at large. So without boring us with so much complexity, I would like to go straight into the topic. So my goal is to further do a sort of orientation, or to in another way, reorientation, because sometimes I know a lot of people that are here, might have one time or one way or the other have an interaction with what bioinformatics is. And maybe some came just to feed their curiosity. So, but to make everyone be on the same pedestal so that we would understand each other greatly and to pass the necessary information that we have to do, that we have to pass, is to define what bioinformatics really is. In the definitions, there are different definitions of bioinformatics, but there are three important components that makes up bioinformatics. And these three important components are ones any country or any continent or any organization or any name it, once they've been able to address these three issues, the study of bioinformatics or even advanced research in the area of bioinformatics would be solved. So by definition, bioinformatics has three components. The first is the computer software or the information technology that surrounds it. The second is the database. And the third part is the molecular biology aspect. And why is that? By definition, bioinformatics, it involves the application of computational methods or computer software or computer analysis to analyze and interpret biological data, such as DNA sequences, protein, or gene expression profiles. Now, in this aspect, the aspect of analyzing biological data, what it used to be was the area of molecular biology, whereby we analyze the biological data, we sequence the DNA using molecular biology method. But as we all know, the first sequence that we have is the sequence of insulin, which is the protein insulin. So, but nowadays, we are now using advanced computer software to analyze the scores, to elucidate and give more information about biological data. And these data are usually stored in a database. So that is where the three components come into place. Once we've been able to address these three components, the issue of bioinformatics is already addressed. Now, moving on to the topic. Now that we've had an idea about what bioinformatics is, now there's a conundrum about Africa being shortchanged, being backward regarding the area of bioinformatics. But how do we address that? What are the problems? Now, the first problem that we have as Africa, because when we want to discuss the need for more bioinformaticians in Africa, we have to understand the problem that we have presently. One problem we have regarding the first problem that I would like to discuss in this area of the need for bioinformaticians in Africa is that Africa lacks the IT expertise. There are lots of, we have a lot of people who are into tech. We have Andela in Nigeria. We have different tech gurus, but there has been little or less attention has been diverted to the health aspect of tech, to the IT aspect, to the technical aspect of information technology aspect of these particular technological advances. Even though, yes, there are some advancements, we have the Andela and the likes, but they are not as popular as we have the Silicon Valley. They are not as popular as we have different pharmaceutical industry that are using IT or computer expertise to address biological problems. We are still going to get to that in the course of our presentation, whereby we can see how bioinformatics has been able to provide solution to the problem of COVID-19. We are still going to get to that if time permits. I have 30 minutes. So there is a lack in the IT sector. There is a lack of expertise in the IT sector that will drive that development in the area of bioinformatics. Yeah, we have a lot of tech, but they are not as sufficient because in Africa, especially in sub-Saharan Africa, we have, in Africa in general, Africa accounts for more than 17% of the global population. But right now, as we are speaking, there is less than 3% of genetic information that is present on the database that are of African origin that is present in the database. Now, when it comes to analyzing or examining these samples to correspond with other samples, we have shortage of African samples. And one of the main reasons we're having that is the shortage of that expertise. Now, another area of the conundrum that Africa is facing is the power. There is little, like we all know the situation, especially if we do not know of any other place, I can say of Nigeria, that most parts, especially sub-Saharan Africa, we are facing the issue of electricity. And yes, we have a lot of talent regarding the IT expertise. We have a lot of talent. Africa is producing them day in, day out. But if you do not have electricity, you have problem because a computer cannot run on its own. That is why you will see a lot of startups coming up with sustainable energy, like the solar, the hydro, the wind energy, just to supplement the electricity from the national grid, as the case may be in each country. But the problem of electricity is a problem because when you are dealing with computer, of course, you have to charge and do a lot of things. I can tell you, as a matter of fact, that electricity can be the #1 problem facing a lot of bioinformaticians in Africa, because there are sometimes you might want to simulate an experiment, and it will require the computer system to be on watch 24 hours just to complete that experiment. But since you are having an epileptic power supply, you might not be able to achieve that goal. Electricity is a problem. Now we have this governmental intervention. That is a major issue on its own. A lot of people do not know that in Africa, most African governments apportion less than 13% of their national yearly budget to health or agriculture. And these are areas in which bioinformatics comes to play: health and agriculture, health and agriculture. But people are not, government across Africa are not giving much more attention. Even some people, when they tell them, okay, we have bioinformatics, they will ask you, what is bioinformatics? I had a personal experience that I told them that, oh, I am doing a sensitization. We are doing sensitization on bioinformatics. They will ask you, is it biometrics that people will be checking their thumb? Like, is it biometrics? I said, we will start to explain to people. There is a vast gap in knowledge about bioinformatics in the continent. I will give a personal example. When I was in Nigeria, that was in 2016, when I was doing my master's program at one of the universities in Nigeria. We were made to go for an excursion or a field trip to another research center in another part of the country. When we got there, it was a molecular biology laboratory. We are shown the electrophoresis, we are shown the HPLC machine, we are shown the nanodrop, we are shown the PCR. But a lot of people that are working in those research institutes do not even know how to operate these machines. And those who know how to operate these machines, they are scared to do analysis or to work on their own because they are scared that even if they press something wrong, the machine might stop working. And that might mean that their daily bread will be stopped because they have caused the damage. So that's issue of that. Yeah, we are in the research institute, but we are not doing the research. It is just for photo ops. So a lot of research institutes like that abound in Africa, whereby the government, if they manage to get the government to do something, they will buy, but no research will be carried out on those using those machines. The expertise to carry out to work on that machine is another problem. We were told about in that same field trip, we were told that there was a time whereby there is a machine, the government bought a machine that they had to send some staff members of that research institute to India to go and learn how to operate that machine. So when those people, they just go for 2 weeks or 3 weeks, those 2 or 3 weeks is not even enough to understand fully the workabilities, the functionalities of the machines. So when they come back, they'll just, okay. And then the machines will just go and it's just be there, or not used. So this kind of situation abounds in a lot of countries in Africa. So that's a problem that we are having. Although there are solutions to that right now regarding training with the use of this virtual training, and we are still going to get to that as to what's COVID-19, one of the benefits of COVID-19 to Africa. So government intervention is still very low. The only intervention that we have right now regarding bioinformatics sensitization, we all know the H3 bionets, and that's the only prominence that we can think of that at the click of a hand, any government, and this H3 bionet is not even a government from African, it's not an intervention from any government. It was a foreign aid that was meant to sensitize and increase the bioinformatics sensitization in the continent. Now, another area whereby the conundrum we are having as Africa is the African Biological Database. There is no, as we've defined what bioinformatics is in our previous slides, there is need for a biological database of African origin, There is an initiative that is currently going on that is being spearheaded by Dr. Ebenezer, thank God. This is an initiative that is working to sequence a lot of genomic data from Africa and maintain this database from Africa alone. So this is an initiative that is privately, from concerned individuals. So it is not a government intervention that, okay, we are commissioning this project, but individuals are coming up with that approach and attempt to ameliorate the problem that we are currently facing regarding our biological database in Africa. Just like I said, that those samples, genomic data of African origin is less than 3% in the global database. Of course, the NCBI that we know, less than 3%, despite the huge diversity of our flora and fauna. This is a major problem that we are having that is against bioinformatics progressing in Africa. Now, another area that I've touched a little bit more is the molecular biology laboratory and expertise. There is a shortage of individuals that are experts in molecular biological technique. Just like I said, we have little facility compared to our population. And yet, even if you are enthusiastic to learn, it will cost you a fortune to learn these molecular biological techniques. Even though these techniques do not need rockets, is not rocket science. When I got to South Korea in 2018, still stuck talking from my personal experience in 2016 when we went on our field trip in Nigeria. So we were just shown all the molecular biology apparatus, tools, non-raw PCR, blah, 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 gene, this, gel electrophoresis, yes, HPLC machine, just look at it. Because we are even scared to touch, because once we touch and something bad happens, we'll be held responsible and it will cost a fortune to do. But this, when I got to Korea in 2018, so I discovered that all these machines, all these tools that companies, institutions, research institutions are boastful of in my country, is if most of them are present in my lab. And this lab is part of a department in my, is a lab in the department. So it's kind of baffling. So all these tools, I was able to play around with them, become expert in operating the PCR machine and other machines that are regarding molecular biology. So I was so surprised that these are things that are missing back home, back in Nigeria, that is curtailing, that is stopping the progress of bioinformatics in Africa. And however, even though to be a bioinformatician, you do not need much facility, you do not need much infrastructure. Just have a good system, have good internet, and have good electricity. You are good to go. However, we cannot take away the importance of the molecular biology part of bioinformatics, because that is when you'll be dealing with the wet lab. Yes, a lot of analysis. We are living in the era of data explosion. There are lots of data that is yet to be analyzed, but still, we cannot leave the part of the wet lab part of bioinformatics. Now, I would like to discuss a little bit about the role of COVID-19 in advancing bioinformatics in Africa. Before COVID-19, I can speak especially for Nigeria in particular. There are fewer laboratories. There are few, we can count, maybe 1 or 2 laboratories that can sequence genomes. I know of some companies in Nigeria that they will collect your sample and even ship them out of the country to another country in Africa to sequence, and then you get the result. That is how bad the infrastructural deficit in the continent is, especially in Nigeria. But with the advent of COVID-19, it's a sad and good tale. We were able to, out of the blue, develop and increase our capacity to sequence genomes, especially of virus of COVID-19. And with our advancement in being able to sequence viral genome, we can sequence other genomes too. That's that sped up the advancement that we see in sequencing. And that is one of the benefits of COVID-19. Of course, when you sequence, you have to analyze. A lot of people, a lot of researchers, I know of different research institutes, privately owned, that are coming up with research that is focused majorly on analyzing COVID-19 in itself, either as a vaccine or a drug candidate. There is a Helix Biogen that is doing wonderful well in that aspect. There is a Genomark and lots of privately owned companies that are doing that aspect. I know Biotech and Omics Logic are also doing amazing in that area of drug discovery. So this has led to development of COVID-19 research, and we are able to have a lot of researchers interested in sequencing and analyzing, and then we are having a lot of African samples from African origin being deposited on the database. And also, apart from COVID-19, it has also led to the development of the metagenomics aspect of bioinformatics. Now, this is just to show us some of the data regarding COVID-19 in Africa. Now, we can see that there is a huge gap between testing that has been reported by WHO and other international organizations on the depth of testing facility to determine the actual COVID-19 cases in Africa. According to this data by Worldometer, it says that in Africa, we have more than 12 million cases that have resulted in more than 200,000 deaths, and about 12 million also recovered from COVID-19. Yes, this is little compared to the population that Africa has. The testing capacity is a problem that we've not been able to determine the exact COVID-19 incident in Africa. However, luckily for us, we do not have a huge death rate to have warranted us to be a graveyard, for Africa to be a graveyard. That's another area of research that I am interested in, that I'm passionate about, that I've been talking about in different speaking engagements that I've been talking about. That why is it that Africa do not have, yes, we have COVID-19 infection, but our death rate is lower, much more lower compared to other parts of the world.
Hello,
Now,
sir. You have 2 more minutes, sir.
Wow. So let me speed up. So this, I spoke about the COVID-19 vaccine development with the help of bioinformatics. What are the African needs? Africa has a high burden of infectious diseases such as malaria, HIV, and tuberculosis. And these diseases, we have the potential to use bioinformatics, especially in genomic studies, to address these problems. That was what led to us writing the correspondence to Nature that got published last year, almost a year now, on the African need small bioinformaticians for population studies. And these has instituted a lot of different research on how do we increase bioinformaticians in Africa. And one of the problems we'll be having regarding bioinformatics in Africa is that, okay, I'm still going to talk a little bit more before my time runs up. Now, the essentiality of bioinformatics in Africa. Africa has a lot of diversity in flora and fauna, but these has not been worked on. We've not exploited our diversity to the maximum limit. The world is now moving to precision medicine to treat a lot of and to characterize particular diseases, and then providing solutions to them require the skills of bioinformatics. And we need that in our continent to be able to move with the world. And one of the problems, one of the challenges is that there is a lack of training programs. And I'm sure that is what Omics Logic is trying to do. And also we from Noble Maths, we are also improving the training, the amount of training that we'll be conducting in Africa. So regarding bioinformatics, and also this led us to writing another paper on African need genomic epidemiologists. So there is need for training. There is a particular research that was done, a particular paper that was published or survey that was carried out by the World Bank that says there is need to, in Africa, to carry out more than 170 million trainings. And COVID-19 has enabled us to move from the class to the virtual world that we have in this session. I've talked a little bit about infrastructure, and one of the problems we're having in Africa is the brain drain. A lot of researchers from Africa are moving from Africa to other parts of the world to also further their research. I've spoke about African Society of Bioinformatics and Computational Biology. These are also organizations that are championing the cause of bioinformatics in Africa, but it is not enough yet due to our population. So what are we doing to complement that effort in Noble Maths? We also have genomics introductory course, not enough, yeah, but it's free. We have also conducted from 2020. We've been doing that, but it's not enough to curtail or to sensitize, but we are trying our best. And we've collaborated with international institutions like EMBL and also researchers from across the world to train African researchers. And we've done different aspects of bioinformatics free of charge. And yet we are having a lot of people joining. So far, our goal is to train 1 million bioinformaticians in Africa before the end of the year. And we've been doing that assiduously from our personal money. This last January and February, we conducted the bioinformatics tour across four universities in Nigeria, and it has been attended by participants from 10 different universities, as well as different research institutes and centers in Nigeria. And this was from our training in Fountain University in Osogbo. We also had a stop at University of Ilorin, where we trained a lot of people. We also had a stop in Babcock University, and it was very, very fruitful. We also had a stop at Crescent University in Abeokuta. Even the Vice Chancellor was in attendance and also applauded our efforts. So thank you so much for having me. It's been a wonderful time. It's been a wonderful session with you. So thank you so much.
Yeah, thank you so much for that presentation. I believe that some persons will be having some questions, and I would love them to actually share that real quick. Yeah, I believe we can all hear me. Can we all hear me?
Yes, we can hear you.
All right, thank you so much for that. At this point, I would love everyone that actually have a question to go to the chat and write the questions. I want you to go to the chat and write the question, yeah, because it's quite interesting. I would also want maybe Dr. Dekunle or Shugari to make me a call so I can share my screen. That's quite important. I believe our next speaker is actually on ground. She would be speaking in a bit. I would be sharing a screen, a slide. We'll be sharing a slide. All right. I believe my screen is visible now, right?
Yes.
Yes, it is.
Yeah. So thank you so much for those lovely input. I want everyone to understand that Africa is progressing, and there is a lot of private and public individuals and organizations that are doing their best to make sure that Africa goes forward. Not only H3 Bionet that got their funding from NHS, and we have very brilliant point that quite connected for us. I mean, it was by Ashraf Akitola, and it was quite a lovely one. At this point, I would love to bring up a reputable associate professor, president of Dr. Foluke Eunice Shola-Ojo, which is associate professor in the Department of Animal Production at the University of Ilorin, and she's a registered animal scientist. Her research centers on qualitative molecular evolution, genomic diversity, studying genetic diversity, performance and conservation of indigenous poultry or other domestic animal species in Nigeria using quantitative molecular and functional genomic approach. Dr. Foluke, are you there? Can you hear me?
I'm here.
So,
good evening. Can you hear me?
Yeah, we can hear you. I will be sharing your screen in a bit, and we can get started. Yeah, over to you, ma'am.
Okay. Good evening, everybody. I hope you can hear me.
Yes, we can hear you.
Okay, thank you very much. I'm delighted to be here this evening to present the topic given to me, and that is the role of bioinformatics in livestock improvement. I've been properly introduced by Dr. Alabetutu. Thank you, sir. All right. I'm going to start my presentation by presenting what it is in my outline. I have what is contained in the introduction, where I discuss bioinformatics and research on bioinformatics, the process of bioinformatics. I won't stay long on this because it has been stated by the last presenter. Then the key of my topic, that is bioinformatics application in livestock production. And I mentioned genomics and proteomics, although there are other aspects of omics, then gene expression, genomic diversity, which is my area. And I move to my conclusion and recommendation, whereby I state the function of bioinformatics in livestock world. All right, let's get to the business. Bioinformatics. I won't go into details of the scientific definition, but I want to define it in a way that a livestock producer will like. I'm going to call it a tools that unlock the hidden treasure for optimum production in livestock. And at the end of the day, it gives good life for man, because when you have quality products on your table, you will be happy and you will feel good. All right. Defining bioinformatics, like the past speaker just said, is like solving biological problem in terms of the DNA, the AAS, amino acid sequence, and other related information through mathematics, statistics, and computer science. You need to be sound in these three aspects: mathematics, statistics, computer science, before you can be a good bioinformatician. And if you are a good bioinformatician and you are a molecular scientist or an animal productionist, it will definitely help in providing information for the masses and for researchers that they can use in improving livestock. All right. Scientific research and bioinformatics. Without research, there cannot be bioinformatics. And bioinformatics actually is something that provides information at the end of every scientific research. As we all know, research starts from having an idea. You have to, put down your proposal, aim, objective, justification, budget. Then you move to materials and method. And at one corner, you are providing information, which you get either through the internet, whereby you write your introduction, literature review, you gather your results for presentation. But for all this to be meaningful and for you to communicate with the world what you are doing, if it is a molecular science research or other scientific research, you need to do data analysis. And under data analysis, you can either use statistics or bioinformatics. In our own case, if it is phenotypic parameter or quantitative aspect of study, we use statistics. But when it's getting to wet lab, molecular studies, we have to use bioinformatics to be able to present to the world what we are actually doing in the laboratory. All right. I don't want to go into the contribution to livestock improvement without talking about omics. It's omics data that makes bioinformaticians to be prominent. Without omics data, what will bioinformaticians be doing? Bioinformatics is the key to unlock all omics data. Genomics, proteomics, metabolomics, transcriptomics, glycomics, lipidomics. If you are gathering omics data, think bioinformatics. Okay, all right. All right. Bioinformatics entails revelation of the unseen. Because if you are teaching in class, it might look abstract to students. But by the time you use the tools, any of the tool of bioinformatics, you will be able to present to the student or to the world what you are actually imagining or what you are saying you cannot see. And it does this by creating advancement of database algorithms, computational and statistical techniques, theory to solve problems arising from management and analysis of biological data. Through your careful planning, careful procedural planning and execution. All right. And this bioinformatics analysis, it's a challenge in Africa and in Nigeria especially. For example, I was just getting ready for this seminar and I was, I was almost ready. I got enough data on my laptop to combine to mobile data, mobile hotspot and use my laptop. My laptop just went off. It wasn't connecting with any network. So this a kind of a problem. And many of us have been in and out of this seminar because of network issue. I have to be moving from one spot of my house to the other to see where I can actually have each free discussion on bioinformatics. So there's a big challenge in developing world most of the things that bioinformatics needs. Bioinformatics need good network with strong bandwidth, good computer with good speed. It needs data, time, maximum concentration. On behalf of bioinformatician, you need to be patient, then resilient. You need not to give up easily. Then you need to be determined that you are going to get results. But we have big problem here. A lot of students, they don't have the capacity to buy data. There was one that I registered, maybe 10 of my students, organized by H3 Bionet. All of them drop off. I asked them why. They told me they don't have money for data. So this is a big challenge in Africa, and it's something that is affecting us in livestock production as well, especially the molecular scientists and the bioinformatician. All right. All right. What are the tools that we use in bioinformatics? Like I said, computer and the internet. Simple. And other software that we use in bioinformatics require computer, from BLAST to analysis of sequence to sequence alignment, editing, gene editing, and all that. You need computer. You need strong internet. So these two things are the basic things. There was one particular training organized for Nigeria, free of charge. They asked us to write the capacity of our computer. They asked us to write whether we have internet facility, whether we'll be able to complete the course. These are very key things that a bioinformatician needs to be able to contribute meaningfully to animal production. Next, all right. All right. We use bioinformatic tools in livestock production we use it to generate information, we use it to mine, we use it to retrieve, we use it to analyze. So when you handle the tools, you can generate a lot of data or information from it. You can mine as in search for a lot of things. Then you can retrieve, then you can analyze from it. All right. All right. What are the bioinformatic websites that we use? Well, I would like to kind of saying, if you want to be a bioinformatician, you must be able to solve the web very well. And I like a comic actor that like, web slinging, web solving, and that's Spider-Man. You could see me putting Spider-Man on my screen. And you should be able to sit down and search the web very well for what you are looking for, so that you'll be able to make meaningful contribution at the end of the day. The website that you can use are, you can use the NCBI, the GeneBank, the Unigene, EMBL, EBI, SwissProt, ESMBL, ISCB, and NCGHub. All these are websites that we have used in one way or the other in livestock improvement. All right. All right. Bioinformatics application in livestock production. Yeah, in livestock production, we use bioinformatics for biological data organization. It gives us access to information, modification of information, medical and health information, diversity information, evolutionary information, phylogenetic analysis. In major research efforts, such as sequence alignment, gene finding, genome assembly, drug design, drug discovery, protein structure prediction and alignment, prediction of gene expression itself, protein-protein interaction, metabolic pathway, genome-wide association studies, and the modeling of evolution. All these, we have a lot of research reports that people have done, and they've positively contributed to livestock improvement from the lab to the computer of a bioinformatician, then to the field, whereby the farmer can make use of it, and it will contribute positively to livestock improvement. All right. In livestock production, I said I want to talk about genomics. Genomics has contributed positively. I'm talking about genomics now. Go back to genomics like that. I have problem with my computer. Okay, yeah, now. Genomics has helped in sequencing of cattle, buffalo, pig, sheep, goat, camel, and poultry, whole genome sequence of this animal. Ancestry prediction has helped in ancestry prediction that aids selective breeding, conservation of extinct animal species, improvement of animals' health and products, understanding genetic diversity, and selection of desirable traits for livestock optimum production. To cap it all, it has done something to the environment. It has helped in sequencing of microbial genome that assists in reduction of greenhouse gas, thus contributing to stability of global climate change, as livestock are the major impetus of climate change in the world. All right. All right. For the proteomics have helped, results have helped in increasing livestock production to give animal protein and ensuring that these animal protein are of high quantity, well characterized, they are authentic, they are safe for consumption. It has helped in detection of microbial problems in animal protein, and it ensures quality that gets to the consumer. Proteomics also has helped the industry by transformation of the industry and detection of fraud. It serves as an eye to see whether a protein manufacturing and processing industry are into fraud, and such company helped in all this. In one day we get there, whereby we'll be using all these tools to do all these things and produce quality products for our people. All right. All right. For gene expression in livestock, it has helped. Tools such as the process such as microarrays, EST, SAGE, tag sequencing, MPSS, RNA-seq, various applications of multiplex in situ, hybridization, and through high throughput gene expression, measurement of mRNA level with multiple new techniques, as revealed in the HTGE, have all helped in livestock improvement. All right. What have we done in my molecular evolution and genomic diversity unit? Well, what tools do we use? We have used tools such as Secma Pro of laser gene, version 7.1.0. We have DNA Star. We have used DNA SP, Mega version 6.06. We have used Aliquin. All these tools have helped us to contribute positively to a lot of information about genomic diversity in poultry species in Nigeria. All right. All right. We have also used, to view our sequence, most of the time, we use JALView. And I could remember a particular time I was teaching in the class. I taught my students in the class mutation, polymorphism, and all sorts of things like that. So it's now got to a stage that they didn't really get what I was saying. And I was like, okay, don't worry. When next I'm doing bioinformatics analysis and I want to view a sequence, I will call you to come and have a look at it. I invited my student after putting the sequence into JALView and for them to see the polymorphism and the differences between the consensus and the main sequence. And I was telling them, look at this. If you check this, you will see that it's different from, it's supposed to be C, it now have 8. It now have A instead of C. And this is part of what I'm talking about, that this animal is different from this animal. And this is what makes it look like as if something has happened to the animal. And this is what gives them the differences. They were like, wow. So is this what mutation is all about? Is this what it is? Oh, I can see the differences. I can see that instead of the sequence to have A, it's having T. Ma, would this be deletion? Could it be insertion? Could it be, omission or something like that? And they have better understanding of mutation. So all these tools, like I said, one of my students now make a comment, Ma, is searching for the unknown, for the seen. I mean, looking for the unseen, and now I can see it. And I said, yes, bioinformatics can make you to see the unseen. So it has actually helped us in livestock production to see the unseen most of the time. All right. All right. We have also used tools like cluster hole and cluster W to construct our phylogenetic tree and understand the relationship, whether it's a monophyletic clade or it's not in animal between our birds, the species of birds that we are studying, the kind of relationship that exists between them. We have used this tool to study it, and we either put all our sequences on cluster O or cluster W and generate the relationship for more explanation and to provide information for the researchers. All right. Now, some of our findings. We have reported genetic diversity and population structure of Muscovy duck in Nigeria, Arena muscata, and we have also reported association of MC1R variation and plumage color diversity of Nigerian domestic pigeon, Columbia livia domestica. We discovered that genetic markers such as nuclear DNA CYP2U1 and mtDNA cytochrome B reveal two groups in Nigerian Muscovy duck population, and also MC1R SNPs, C206T, G253A, and A520G. They were informative in Nigerian pigeon, and the SNPs were assigned to 7 haplotypes, and 2 haplotypes occurred in all the plumage types. All right. All right. Now, our findings have been reported in reputable journals, and we have been able to contribute to knowledge from this study and other studies like that, but I just picked these two because of time. Our studies have shown that there's no genetic differentiation between and within Nigerian Muscovy duck. Human activity shape population structure of Nigerian Muscovy duck, and it shape it because people that they give them out as gift. People travel from one place to the other and they carry their animals along. Our study also revealed the suitability of DNA CYP2U1 gene, and it provides insight into the variation in MC1R and plumage color diversity association of Nigerian domestic pigeon. All right. Now, my conclusions and recommendations. There are other aspects of livestock that researchers in Nigeria or Africa need to continuously, especially molecular scientists in Nigeria, need to continuously be using bioinformatics tools. From their wet lab, they should use the tool to generate information that will be useful and improves our animal further. Bioinformatics tool in livestock research has provided more information on DNA sequences, genomes, gene variation, gene regulation, gene expression, and mutation. You have to embrace omics and bioinformatics research to understand livestock. Then you have to do omics and bioinformatics research to make livestock production better. All right. All right. Use of bioinformatics for livestock improvements can be through analysis of biological data, gene structure, protein structure, gene expression, gene regulation, and gene variation. Bioinformatics makes livestock production easier and better. All right. Use of bioinformatics in livestock research has been useful in prediction of gene structure and function, protein structure and function, gene regulation network, simulation of the environment. And you have to make your livestock happier, and you will get quality and quantity protein for consumption from them, which will make life easier for people. All right. Use of bioinformatics has been of great importance in detection of gene regulation network, simulation of the environment, mapping and aligning of DNA sequences and protein sequences. And when you use all these things, you're going to produce livestock that are fit and viable. All right. As a scientist, as a molecular scientist or wet lab scientist, I want to state here that you have to be a bioinformatician. And when you are a bioinformatician, you are going to be happy, you will stay informed, you will keep informed, you'll be a model, a solution provider, you'll be knowledgeable, you can make money and you get busy all the time. Then you provide scientific information, you'll be a star and you'll be an oracle. You know why I have to put an oracle is this. In Africa, is if you want to see the unseen, you consult an oracle, then the oracle will tell you that this is what's going to happen, but this is how it's going to be. So there is even there was a time I enrolled in learning more about oracle and how to use oracle, and I was very happy at the end of the day. So as a bioinformatician, you just be like an oracle because you're able to provide solutions to most of the information or things that are happening in the world. Now, my recommendation. Creating awareness on omics research and bioinformatics analysis in all African University and Research Institute is very, very important. It's germane. It's something that we need to do. We have to think bioinformatics all the time. We have to keep creating an awareness that people should engage in bioinformatics for not only livestock production, in health, in food production, and in everything that will make life easier for everyone. For the youth, I want to encourage them that they need to be a star in omics and bioinformatics study. You still have plenty of time. You still have a lot of things to do that will make you to contribute significantly to livestock production and other aspects you are, whether you are a medical student or whether you are anywhere you are that uses omics and bioinformatics. Be engaged and try to know more about it. For the adults, I want to encourage you that age is not a barrier. You can start today. Once you are determined, I started bioinformatics at the age of 45 or 44, 45. And I was the youngest in my class all the time. And when I started with my second training was with H3 Bionet, I used to write notes and they would be looking at me, Mommy, you are writing notes. I was taught to write notes. And I thank God today that I can say that I've used a lot of the tools and I understand them. So age is not a barrier in bioinformatics study. Also, livestock biological data in Africa needs to be upgraded to omics and bioinformatics level for optimum production and provision of adequate protein for the teeming population with ease, because all form of omics contributed to livestock production, and you can't have any of the omics data without using bioinformatics too. When I started, I thought I'm just going to be a molecular biologist. I just have biological data. Then I discovered I have to be paying heavily to do bioinformatics analysis. I said, then I need to go back to class. So I had to go back. I have to sit with them. Most of them, the first time in Spain, when I was in Spain and they gather us together and that we should come and have a lecture on bioinformatics, we should open our computer. My computer doesn't have the speed and the capacity. I sat with a very young fellow. She was very kind to me and she put me through. So we need to keep on doing this so that a lot of people can have, the knowledge of bioinformatics and they can impart it to the younger generation. So to this, I've come to the end of my discussion. I thank everyone for listening while I wait for questions and contributions. Thank you very much. These are my references. Thank you very much. Thank you for listening.
Wow, thank you so much for those. They're quite on point. As a population geneticist myself, it was quite interesting to see how that the same tools I use for my own research are also the same tools that you use for your own research, and it's quite interesting and enlightening to actually see that. Thank you so much, Dr. Professor Foluke Sholaujo, for that. It was quite interesting. And I want to enjoin us to get ready and get set because another section will be coming on now. And please, if you have any questions, feel free to write the questions down in the group and we will be throwing the questions out. All right, so that's Dr. Sholaujo again for you. I believe we can all see my screen, right? I believe you can all see my screen, okay?
Yes, we can see it.
Okay. All right. So we have met our speakers, and permit me to mention that we'll be having a panel session right now. And as part of this panel session, I'm convinced that our panelists will be doing justice to the discourse around bioinformatics education, training, and research in Africa. Many African countries, just like Nigeria, are also facing the same issues that Nigerians also face. And I must actually say that because we engage universities from Ghana, from Cameroon, from Yaoundé, and co. And we know that we are all in this together. So Africa is quite a place to be in engaging some of these subject matters. We have met our several participants. We've met Ashraf. We've met Dr. Foluke. We have not met one of our last finalists. He's a senior lecturer. I believe is around now. Dr. Abdulaziz Anjorin, are you around, sir? The panel section is about to start. Hello, doctor, are you there?
Yes, I responded. Thank you very much, Ayonfe. I'm happy to be with you.
All right, thank you so much, sir. Let me just introduce him shortly. He's a senior lecturer and medical biologist at the Lagos State University Department of Microbiology, where he leads research on influenza and other respiratory tract viruses. He graduated from Lagos State University, his PhD from the University of Lagos. He has been trained by the WHO Reference Laboratory at the Luxembourg Institute of Health and the International Society of Influenza and other respiratory viruses. He has conducted groundbreaking research on influenza and other viral infections and presented papers in diverse countries, won numerous awards, and is a member of several professional societies. So, Dr. Anjorin, you're welcome again. It's so lovely to have you here. We also have another panelist, which is the president of our program director. We've actually met him. I have spoken to us at the beginning. I would love to say that he's such a wonderful person to actually work with. He's had a more than 17 years combined experience cut across public and private diagnostic practices and so on. He's a data enthusiast such as myself, and I would be asking some very, very typical questions. So I would love all of our panelists to make ready themselves. Dr. Ashraf, Dr. Shola, Dr. Adekunle, Dr. Anjorin, I believe we are ready and set. So real quick, to start a question, I would love to begin by saying that Africa is a home for some of the most diverse and biologically rich regions in the world. However, many African countries are facing significant challenge in healthcare, in agriculture, and environmental management. Bioinformatics, like we have heard today, can play a very crucial role in addressing these challenges by enabling researchers to make sense of complex biological data, developing innovative solutions. Yeah, we've also heard and seen so far that bioinformatics is rapidly growing field. It combines a lot of these different fields for us to analyze and to interpret biological data. And it's becoming a very, very important and essential tool for researchers in the life science, and it allows us to process and analyze large amount of data efficiently. But in Africa, the need for bioinformatics education is particularly acute. It's particularly acute. So I would love to throw the first question to Dr. Anjorin. In your opinion, the need for bioinformatics education in Africa is urgent. So can you actually share from your own opinion, seeing that you've actually you are cross-sectionally experienced with different continents? What will you say is the current state of bioinformatics education, training, and research in Africa, sir? Dr. Anjorin, the
Okay.
question to you, sir.
Yeah, thank you very much. Can you hear me and can you see my face?
Yeah, we can hear you, sir.
Okay, so I think I must commend you and to also appreciate your company as per what you guys are doing in Africa. Permit me to also appreciate the previous speakers, especially the last one, our associate professor, Dr. Foluke from the University of Ilorin. So to start with, you did mention about the importance of bioinformatics to agriculture in your opening remarks, Ayanfe. But let me just quickly correct that we've gone beyond that now, and that the entire world is now looking at the application of bioinformatics at One Health. We had a global congress at the World One Health conference in Singapore lately, last year, where a lot of things, were said. And now what's the whole world, the World Organization for Animal Health, the World Organization for Animal Health, the WHO, the World Health Organization, the United Nations Environmental Protection Agency, UNEP, and several other international bodies, what they are all looking at now is the study that has to do with
I think Dr. Anjorin is breaking now. I think Dr. Anjorin is breaking now. All right, I think I will have to call on Dr. Foluke to
animals and of course the environment with
the connected
and I can also tell you, hello, can you hear me?
Yeah, we are following now.
Okay, I said with a lot of effort so far in the 1970s, in 1980s, about nucleic acid, about protein, and their analysis, and with the coinage of the world bioinformatics in 1989 by Daniel, Massis, that we can also talk about the first sequencing of the Haemophilus influenza microbial entity, in 1995. That is the first genomic sequencing of any microbial entity. To also talk about the 2001 human genomic, first publication, and to narrow it down to Africa. A lot of things are currently being done, and I can tell you for free that Africa is also, putting our best as far as bioinformatics, is all about. It's just that we need to do more, as regards bioinformatics in the entire continent. But I can tell you that we are not doing bad. We've picked up. Before now, we've been slow. We do not know what bioinformatics is all about. But I can tell you gladly that now, Africa, our stage, and we are gradually coming into, bioinformatics and what bioinformatics is all about. So that is the introduction. Over to you.
All right, thank you so much, sir. Let me call on Dr. Foluke straight away. From your own first-hand experience with lecturing, now talking about bioinformatics education in the university system in Africa, what will you say the experience is? Is it actually at the university system, are they adopting it, or it's actually an independent race? Yeah, so that's the question I'm trying to you, Dr.
Okay, thank you very much, Dr. Anyofe. That's the question. The question is in two ways. In some university, they've taken it up and they've added it to their course system, whereby a student of biotechnology, molecular science will be taught basic topics in bioinformatics. But in most universities, even my universities, it's an individual race. Like the first doctor that spoke said he came to train some people in the University of Ilorin. I'm sure maybe it is their supervisor that organized it. I don't think it is the university that organized this for the students. So it's an individual race. In my own case, it was an individual race. And when I was spending money to actually leave quantitative, aspects, all these diet manipulation and other things, and face molecular and bioinformatics analysis, I have to sponsor myself, train myself and pay for it. So in
All right, I think we are losing Dr. Fuluke there. Yeah, but the message is quite reached. She's saying that it's more like
every university we embrace it, put it in the course system, from the basic of bioinformatics to the rudiment of bioinformatics and everything. Thank you very much.
All right, thank you so much. I will be going to the next question, and I will be calling on Dr. Ashraf. Dr. Ashraf, you are not presently in Nigeria or in Africa, I suppose, or are you? I would love to ask. Okay, sorry, did you say yes or no?
I'm not in Africa presently.
All right, so the question I would love to ask from you now is kind of in comparison, and that is actually about, from your own perspective, research-wise, how do you think educational institutions in Africa, how do you think they're utilizing bioinformatics in improving? How do you think educational institutions in Africa are utilizing bioinformatics in improving the quality of their teaching and learning? Now, I'm not talking about research. I'm talking about teaching and learning in the continent, because there is one thing to teach bioinformatics. There's also another thing to actually learn bioinformatics. Do you actually think that the educational system in Nigeria are utilizing bioinformatics system itself to actually teach? Yeah.
Thank you for the beautiful question. In my interactions so far with a lot of researchers from Africa, before I double down to Nigeria, I feel that there is, especially in countries like Kenya, Tanzania, Uganda, and of course, South Africa. These are countries that are competing with Nigeria in terms of bioinformatics teaching and even in its research. So I can say categorically that Morocco, Egypt, these are also countries that can also be included on that map. So of course, Nigeria, South Africa, Uganda, Kenya, Tanzania, Egypt, those are the countries that are at the forefront of bioinformatics teaching. So in terms of teaching in Nigeria, I think a lot of people, a lot of lecturers in this case, they can teach, they have the ability to teach, because one thing that we are very good at in Nigeria is that theoretical aspect of things. And that is what makes bioinformatics easy. However, a lot of people do not understand the rudiments. I think that is what makes it difficult to understand regarding bioinformatics. So, but teaching, from the researchers that I've been meeting so far, there is still a huge gap in terms of that ability to teach, because there is an Arabic saying that says that you cannot give what you do not have. So, because right now, my major focus, as you can see during my presentation, was our target at Noble Gimat was to target the teachers themselves, the lecturers, because once we've been able to pour to the lecturers, they'll be able to spread it to the students. And right now, to allude to what Professor Fuluke said regarding the bioinformatics in the universities, I heard when I was in Nigeria, especially at Babcock and Crescent University, and I later confirmed from lecturers from Fountain University and that of Uniloring, that the NUC has already added bioinformatics as a core course as part of the, I think, CC Marks or something. I didn't get it is a curriculum. So the National Nigerian University Commission has already added bioinformatics to the teaching. Like it's a core course now. Bioinformatics is now part of must be taught as part of the courses to be taught in Nigeria. So it's a progress, but regarding the present teaching ability, I think it's still very, very low from what I have seen so far compared to other countries in Africa and also in other parts of the world.
All right, thank you so much for that summit. I would love to call on Dr. Adekunle now. Dr. Adekunle, I would love you to speak to your own experience firsthand as a lecturer, your experience firsthand as someone that has worked with the Omics Logic team, your first-hand experience as someone that has worked with the code playground, your first-hand experience as someone that has actually interacted with the Omics Logic learning platform. What do you think the bioinformatics education and teaching strategy, what do you think they can do better? In terms of bioinformatics teaching and learning strategy, what do you think they can actually do better in the Nigerian teaching or in the African teaching environment? You are muted, please.
Okay, great.
Yeah, we can hear you now. Yeah, we can hear you.
Thank you for that question. I want to appreciate all other speakers and their thoughts on this question. In my own opinion, from experience, I've seen that before the advent of the systems, which I'm going to speak a little more into, you notice that it has been an individual race, personally by individuals from institutions. And for some very few institutions here in Nigeria, we have seen some level of adoption at either departmental level or faculty level. Maybe one or two institutions that is actually embracing bioinformatics at the university level. Most importantly, they are the private institutions. And for some of the universities we've attempted to work with, we've seen different, reactions from the stakeholders. I can share that there are some instances where the students will feel, since it is not part of our core curriculum, then they see it as an extra load of work for them. Whereas there are some students who, having the opportunity and getting to use or learn bioinformatics, get excited and they want to actually go on deeper and deeper, to learn bioinformatics. So, but however, I'm happy with the new core curriculum, and minimum academic standards known as CCMAS by the National University Commission. Okay, and with what they are doing, they are actually providing 70% of what should be taught, giving about 30% of that curriculum to universities to develop. And I think this is where I will actually be calling on, researchers, faculties, and, university management to actually use the opportunity to build and inculcate bioinformatics into their educational, curriculum, because the NUC is actually expecting institutions to build 30% of that curriculum. Dr. Ashraf mentioned that so bioinformatics have been inculcated, yes, but then when you look at the skill sets of individuals expected to teach these bioinformatics, then you know that we are up against we are climbing an uphill, okay? But nonetheless, it's a good beginning and it's a good one that the NUC is now giving a framework to allow bioinformatics to be inculcated and included in the curriculum, knowing or hoping that most of our institutions, our university management will now begin to invest in these training capacities and particularly working with international platforms and collaborations that will help scale up the training of capacities, both for lecturers and researchers within the university and also students to be able to take up bioinformatics education within our system. So for me, it's we are at a tipping point here in Nigeria, and I'm happy to hear that most institutions across or organizations across African continent are actually lauding and appreciating the CCMAS designed by Nigeria. And so we hope that they also follow that pace, and that could perhaps provide a good good tipping point, a means of scaling at a more broad perspective bioinformatics into our education and research in Africa.
Yeah, thank you so much. I would love to weigh something in before I call on Dr. Anjorin in the bit to talk about scientific community in Africa. So I would just love to just add one or two things. As a bioinformatician myself, I would love to say that the know-how of a bioinformatics of a bioinformatician user, the bioinformatician consultant, of a bioinformatics engineer, and the other aspect actually differs from one another. I believe that that the universities are kind of breeding bioinformatics users, people that actually knows how to use it. They are not actually, they're leaving out some other aspects of it. So we should be careful as educators to leave out the other aspects. It's not so cool when you teach people how to use an application and they don't in the future know how to make the application, to produce the application, to become bioinformatics programmers. To be able to produce what you really want. Many of these aspects are being dominated by specialists from other fields who actually do not know the basics of what you are trying to get to. You actually have to explain to computer scientists what your aims are, and in the long run, time will actually go. So there's actually a need and a call for even these educators to start digging how to start becoming multidisciplinary. That's one. Then secondly, speaking to our code.omicslogic.com, there you don't need to actually know how to code, but you can just go there and look at those codes and start building your own code. You need to start building your own code blocks. Yeah, that's an advice I would love to give everyone. Actually go out and start building your own code database so that in any case you would be able to not be thrown off board or off target because someone is mentioning something in Python, someone is mentioning something on R code and so on, and it will not actually sweep you off your feet because you understand what the basics are. We are biologists. We have very, very good grasp for science. So at that point, I'm going to stop. And the next question will go to Dr. Anjorin. I have actually noticed that Dr. Abdulaziz Anjorin is quite a community, is a scientific community person. Several times, even before I reached out to him, I've seen a lot of programs we actually anchored as a host. I actually joined. I actually did not tell him. I did not mention to him that I was part of some of the communities that he actually hosted. Yeah, and where he was a moderator for some of these communities. Yeah, that's actually because I'm also a part of the scientific community and I'm a part of a lot of scientific community. So Dr. Anjorin, that's actually a shocker for you. Yeah, but so the question now goes forth. What step do you think can be taken to foster a vibrant bioinformatics community in Africa? You can take it to Africa. We can take it to a sub-level to Nigerian. We can still take it to a lower level, Southwest.
Okay, thank you very much. So that means I'm going to charge you, for joining the the programs, without reg. So thank you. That's a beautiful one. To start with, let me just correct an impression. I appreciate the earlier speakers that it is true that we now have the CCMAS. In fact, we just wrap up that of my department. And we are now about submitting to the faculty. And I happen to be one of the experts that were selected to be part of the committee that wrote the CCMAS for my department. But I want to add to it that even before the advent of the CCMAS, the bioinformatics has always been there in our curriculum as given to us by NUC, that is the National University Commission. But unfortunately, the challenge has been raised by some of the earlier, speakers, that we do not have enough resource persons to, gloss over or to take the course, itself. It is part of courses, but you find out that lecturers in the department, we just keep that synopsis, that part of the, of the course content without, going over the bioinformatics. Until recently, when we started having some of the colleagues, who traveled abroad to acquire PhD in bioinformatics and all of that. Now, to come back to what we are doing at the scientific community level, or what can be done to, resolve some of the challenges, I can just tell you straight away that the solution is one of the things you guys are currently doing. I must salute the Omics, that is the Omics Logic, Africa, and Omics Logic, generally. I've been part of the company before now, learning a lot of things from them. I think the team based in the US and several other countries, and to also talk about the Omics, Logic that is based here in Africa. So you guys have started the work already, and I can tell you a lot of other bodies, the African Bioinformatics Network, ABIONET, the South Africa National Bioinformatics, platform, the H3 Bionet, a lot of them, the Nigeria, bioinformatics group as well. They are already doing what's meant to be done. But the fact remains that we need to do more. Can we go down to the community level? Can you guys now group up, come to each of the departments, come to faculty, call on the scientific community within each of the Nigerian university, like Dr. Ashraf said, that now is willing to partner with some of the institutions, to train the, it should be a program of train the trainers. The experts, the lecturers are the one that will in turn take the postgraduate students, the undergraduate students. Can we have more of the PhD students, running bioinformatics as a program in all of our universities? That are interested? Can we have the, bioinformatics, being taught at postgraduate level? Can we have workshop, training for the lecturers in the biological sciences, in computer science, in mathematical, in physical sciences generally? Can we have a platform where all of the team can meet? Like I previously mentioned, that we have a lot of studies to be done at One Health. Like the advent of the COVID-19, the SARS-CoV-2 needed to be sequenced. We got a lot of experts coming on board, Professor Happy, Professor Milabu, and the rest of them. But can we have more? Can we have a lot of laboratories across the entire Africa doing more? Apart from the pandemic that the government, the government of Africa is willing, the CDC and the rest of them, can we do common surveillance for viral, influenza in birds, the avian influenza, at community level, at the farm level, in the hospital settings? But for us to have all of that, it means we must do more of the training, like you guys have started already. We must do more. Are we willing to write more books? Like I was reading, I read a book, a publication by Oxford, publisher, by Bishop et al. On bioinformatics in Africa. Can we have more books published about bioinformatics? Can we do more training? Can we do more workshops? And a lot of other things to be done within the continent to ensure that surveillance and several other assignments can be done within the scientific community. So over to you. Thank you.
Yeah, thank you so much for that challenge. I have actually been enjoying this session because I see everyone bringing their expertise to the discussion panel, and it has actually been a very, very rich one. I would love to bring the session to a close by asking this last question. And the last question is about what measures do you think are needed by the African countries to capitalize on the potential of bioinformatics for economic development. Now, can bioinformatics serve the purpose of economic development? If yes, I mean, knowledge of or the applied bioinformatics, can it serve the purpose of economic development? If yes, because it's actually the essence of the economic importance of bioinformatics as a subject is the only reason why the NUC, the union, I mean the National University Commission, that's the only reason why they can add it to the curriculum in the university. It actually means that they have bioinformatics actually have what measures, please just have only 2 sentences to make or 2 points to make. What measures do you think are needed by the African countries to capitalize on the potential of bioinformatics for economic development? I'll call on you, Dr. Ashraf. Are you there? Hello, Dr. Ashraf. Are you there?
Okay, yes, I am. Can you hear me now?
Yeah. Please, yeah. Now we can hear you.
That's a very big question, but difficult to summarize within a few sentences, but I will try. Number one challenge I see that, of course, the bioinformatics has tremendous potential to improve the economic situation of Africa. We have that tremendous potential. But before we make use of, start talking of the potential, there is this problem, and it's a perennial problem. Professor Foluke has already mentioned that, and that is part of data. The data is very, very, very important, very slow in major part of Africa. The bandwidth is very slow. Secondly, the good computer system. So, and that is the reality for a lot of people that we are kind of sensitizing, that, oh, come and do bioinformatics. They will say they don't have enough data. If they have, the bandwidth is very slow. And if they do have the fast bandwidth, yes, they have their computer system, they do not have the capacity to carry out that sophistication. So I don't know how we are going to do that, but the tremendous potential abounds so much. I love the presentation of Dr. Abdulaziz on Dr. Anjori on the efforts that have been put in place to improve bioinformatics in Africa, and the efforts that Omics Logic and other private or public funded institutions are doing. But we have a perennial problem. And once we've been able to solve that problem, I can assure you that the issue of bioinformatics in Africa would not be any issue any longer. So we have
Thank you so much.
Problem first.
Like Dr. Foluke said, or Professor Foluke, like I would love to call her, she said every molecular scientist should be a bioinformatician. I took note of that. It has actually sticked with me. So I would love to call on her to ask the same question. What measures think African country can take to capitalize on the potential of bioinformatics?
Thank you very much, Dr. Anyafe. Actually, Dr. Ashraf have said everything, but I'm going to add that, bioinformatics will actually lead to economic development in Africa if the government now, on their own part, have decided to start funding research that are bioinformatics based. At times, when you write a proposal and it doesn't lead to food on the table directly, you hardly get anything. They look at it as if it's full of mathematics or something like that, what food does it want to contribute to the table? You might want to search for something that will be useful in future, and you need bioinformatics too. Researches that are tailored towards this aspect should be funded, so that a lot of researchers will have ability to do molecular research that will end up in bioinformatics. Quite a lot of researchers cannot do molecular research. In Nigeria, it's not all the researchers that are being funded, and a lot of people cannot use their money to fund research or to do research. So the government needs to do this on their own part. Also, the scope of research that will involve the use of bioinformatics too should be our objectives and aim as a researcher too. We should be focused and we should develop more of this research. Then the more bioinformaticians need to be trained. That is through train the trainers that Dr. Adekunle said. They need to train the trainers so that a lot of us will have more knowledge on bioinformatics. Then we can train the postgraduate students and train the undergraduate students on bioinformatics. And to cap it all, good computer, good data, money, finances, and all sorts of things. We actually make bioinformatics education to be prominent and to be embraced by all in Africa. Thank you very much.
All right, Dr. Adekunle, real quick. Yeah. Thank you so much. Yeah, Dr. Adekunle, real quick, can you speak to the Bioinfo server? What solution is it actually bringing to the people, for example? And, okay, yes. And you can just link it to the measure that is needed by African countries to capitalize on the potential of bioinformatics.
Okay, yes, thank you very much. I want to appreciate the earlier speakers. I would also conclude with them that it is going to really contribute to economic development in Africa. And so what we need is to talk about measures and these things that we need to put in place. We talked about infrastructure. Investing in infrastructure is very key. And we need to, find a way to speak more, engage with our government representatives, and to invest in infrastructure. And our hope is that most of this investment will also not be misplaced. All right, we heard lately of the government here in Nigeria wanting to put internet at what is it called now, airports. All right, it would have been more laudable if we heard that, oh, a government wants to put internet in all Nigerian universities. You can imagine the impact, the effect, what that will contribute. But of course, they're after their.
Hello, the universities are included too.
Okay, great. Thank you for that information. All right, so we really need to invest so much into infrastructure, good internet, not just putting something there that cannot download anything. Take, for example, in my own university, we have internet, but I've come to realize, just like Professor Fuluke mentioned earlier on, at some locations, the internet is not as good compared to some other locations. And so you might have to eventually still fall back on your own personal internet, data and all of that effort. So we really need to do much in that aspect. And what I will add to speak to your question. So in Omics Logic, we actually have the Bioinfo server, okay, which helps to take most of these pipelines and bioinformatics activities, explorations of the researcher or the users doing them on their local machines, but to rather do them on the cloud server. And so that's one of the ways we think we can be able to contribute to that aspect of infrastructure deficit. And I also want to encourage that our government or institutions look into that. We can have institution-based data centers and or a cloud services provider or higher computing machines that can help improve these bioinformatics in our environment, knowing fully well that the output will remarkably improve the economic potentials of our country. And lastly, please, there's also need to begin to look at entrepreneurial aspects of bioinformatics. I think that is one other aspect with which we can, of course, this will involve partnerships between the industries and the academics, okay, the academic environment. But we need to also begin to look at that as we might see more and more input or output or empowerment, economic empowerment from that perspective. So I think I want to yield my mic at this point. Thank you very much.
Yeah, thank you so much. There are some questions that are coming in. I should also ask this question, throw this question to Dr. Anjorin. For 1 minute, I would love to just pass a question. Pass a comment, and the comment says that Okay, I can see Dr. Azan's hand. I will call you in a bit. I would love to pass a comment, and the comment is that even in developing countries, for example, not every institution has the opportunity to have a server, for example. So what we do in an institution where I am is that we go with a memo, with our own institutional memo to another institution that has a stronger server that can actually do our same work, for example. And we'll go there to go and write that, we will be using your server for these next 2 or 3 days. If they can't give us, because we are going to have an interrupted data process, they will tell us that, we can only afford to give you for 1 day. Actually, sometimes we also pay for the server access. So, but if it goes from an institution to another institution, it's going to be subsidized. So this actually brings and gives us a challenge, and the challenge is cloud, not just cloud computing, cloud container making, and some of this server, and serious server hosting, because that's actually one of those resources that I think that Africa needs to actually capitalize on. So on this note, I'll call on Dr. Anjorin, then I'll call on the hand raised, then I will also call on the program director after. So Dr. Anjorin, can you still speak to the measure that you think that the African countries, or maybe local authorities of universities, maybe local government of the state can actually take to capitalize on the potential of bioinformatics for economic development?
Okay, thank you very much on that. Yes, everyone is saying the government. I quite agree that the government should do more, but the government is actually the people. One, there must be more commitment on the part of the individual. Myself, yourself, and everyone, like we've been doing. We need to do more. Can we do more? That's the question. Then number two, like it is done in other countries of the world, and that is the position of our union, the ASU, that every company, the private institutions must come, to the fore of these challenges. Somebody mentioned the issue of slow internet, capacity, of the system and all of that. The companies that are making millions of dollars must be taxed. The Dangotes, the Unilever, the Liver Brothers, and the rest of them, they must have a certain percent of their profits, remitted for bioinformatics or for higher institution training, research funding, and what have you generally, to the point that, when we have all of that put together, we can say this is a percentage that is going into medical science, that is going into accounting, going into all of that, including bioinformatics. And when the people are well trained, we are not talking about lecturing now. It is not the issue of the NUC bringing bioinformatics as part of the content to be lectured in the university, but rather it should be taken as a skill, as part of the skill sets to be acquired. There must be workshop, there must be hands-on, technical training, so that people acquire the basic skills, and at the end of the day, they can do something meaningful, design better projects that will eventually translate into money, including vaccine production. We needed to produce vaccine, during the, still going COVID-19, the monkeypox is around us, the Lassa fever is still very much there, the Ebola in Africa, and all of, and all of that. So we need to design vaccines for all of these diseases. So to do that, people must be well trained. Then they now design projects that will eventually translate into, money making for each of the countries in Africa, and Africa, as a whole. And to narrow that down to the last point you raised, that, what can we do? Of course, a lot of things can be done. And once we start from the issue of training ourselves, the individual, getting committed, being interested by themselves, the government cannot do all of that. People, the scientific community must be interested, like a lot of them have said. They started training themselves. Myself as well, I have to start joining different groups, learning it, going abroad, sponsoring myself, getting a little bit, from funders, applying for grant. And so at the end of the day, it is when people are willing to do it, then we now talk about the government, the private sector, and all of that coming into this, the system to help. And at the end of the day, we end up having people that are well trained that will do something meaningful that will eventually translate into money making. So over to you. Thank you.
Yeah, thank you so much for that. We are saying and we are clamoring for bioinformatics to get into the industry. Like the insight Professor Shola Ojude shared. I mean, these are money making. I mean, avenues really. When you have quality products, we have quantitative products, and they are still up to quality. Yeah, I believe that is quite good. There are two questions. Before I read the two questions, I would love to call on W. Akin Azan. I believe you have a question. Your hand is raised up. Are you there?
Yeah, I'm here.
All right.
Yeah, thank you very much for the opportunity. Can you hear me?
Yeah, we can hear you.
Yeah, thank you very much for the opportunity. I want to congratulate the organizers of this program. They have done very well. And also, my gratitude will also go to the speakers as well as the members of the panel. Someone asked a question. What do we do to really make bioinformatics relevant to livestock production in Africa? I think if we want to achieve that, we need to pay attention to animal performance recording. Bioinformatics requires data, and the organism, the mechanism for data collection on animal performance in Africa is still very, very rudimentary. So I want us to take that along. Without data, we cannot make good advantage of bioinformatics. I also need to appreciate Professor Shola Ojo for her brilliant presentation. And I like her advice for the adults. It's not easy to really fall in line with new technologies. I remember my professors in Germany had to learn computer at very old ages, and they made very good use of the technology. So I don't see why old lecturers in Africa cannot also be brought online in terms of bioinformatics. I appreciate Omics Logic, and I promise you, as a representative of biotechnology on NIAS Council, that's Nigeria Institute of Animal Science, that if chance is given, we would like to collaborate with Omics Logic as far as biotechnology, and especially bioinformatics and other omics are concerned. So thank you very much for giving me this opportunity. Thank you.
Thank you so much, sir. We really appreciate the feedback you've brought and so on. I believe that you can reach us through Dr. Shola Ojo, and okay. That is that. Yeah, in fact, we work together a lot. And the next question goes to, thank you so much, sir. The next question goes to Dr. Adekunle. I believe this question also, I mean, Dr. Anjorin may also be asking the same question. Okay, I know why I actually passed that comment. Dr. Adekunle, here's the question. It says that you had training across universities in Africa, because we heard you mention some of them, of course, yeah, but in Ghana, Yaoundé. So what were the criteria for choosing those universities? That's one. Secondly, what are your plans for other universities in Africa, especially in Nigeria, with respect to bioinformatics training? Yeah.
All right, thank you very much for that question. I believe you can hear me.
Yes, we can hear you.
Hello?
Yes.
Okay, great. So thank you for the question. Yes, we've run some programs, particularly with Ghana, a lot of them here in Nigeria, of course, Yaoundé, that is University of Cameroon, Yaoundé, and several other institutions like that. So our criteria, we do not have a criteria to actually select an institution over the other. It's just availability. At times, some of those collaborations come from our programs where we have participants who participated in our training, and then they are willing to open that vista of opportunity with their own institution. So with just an introduction, we're able to move on with such a partnership and exchange communications, and we end up carrying out such a training. And equally, I think I want to state here on this program, we are open to collaborations, we are open to partnerships. Our goal is to just create the avenue for many researcher students, postgraduates, undergraduates, as well as researchers, faculties to have opportunities to use these tools or have access to these tools for their own researches and learning or training. So we are open to these collaborations. But like I said, most of those trainings, recently we have some collaborations opportunity coming up with University of Port Harcourt, as I mentioned. And thankfully, we have one of our alumnus from that, that is a lecturer in their university. And so when the opportunity for their setting up their research, biotechnology center came up, she reached out, and so we've been on that together. So that is essentially what we've been doing. However, I'd like to say we look forward to seeing more and more institutions from Africa joining the Omics Logic platform. If you come to our platform, you see a whole lot of institutions in India, in the United States, institutions, universities, leveraging on the resources of Pine Biotech, of Omics Logic for their own training. Okay, University of Georgia, for example, as part of their training, their postgraduate program, there is a part of the semesters where their students have to take some courses on Omics Logic platform. So why not? Same thing, our universities can leverage on this, starting from our biochemistry students, biotechnology students. The university can simply allow them take these courses either as graded courses that will be transferred to their their grade points, or even as just programs they can take just to improve their capacity personally. So these are the things Omics Logic Africa and the Pine Biotech with our learn.omics platform is available for, and we look forward to forging such partnerships. So I will end by appreciating I think Professor Dr. Akin Azan, who spoke earlier on. So yes, we are open to such collaboration with your institute or our association. Please, we will not mind having your email. If you can drop that for us on the chat, or perhaps you can write us at africa@pine.bio so that we can take further these collaborations talk. Thank you very much.
All right, sir. Thank you so much. Saying a big thank you to everyone that has been on our team. Yeah, starting from Dr. Ashraf, you've been so wonderful. Professor Cholaojo, you have been so lovely having you around and seeing you again. Same as you, one of my mentors. Dr. Anjari, it's so lovely seeing you after a very long time. And it's so lovely. I've also been having the program director here for Omics Logic Africa. It's so lovely to have been working with you for so long. You've been so lovely and wonderful. Thanks so much, everyone, for joining us for this session. Before I call on the next section to take over, I would love everyone to on their camera. I would love to take a screenshot. I would love to take a screenshot. So if you are there, if you can hear me, I want you to turn on your camera. I would love to take a screenshot. Yeah, I don't know. Can you hear me? Yeah, thank you so much for those that are turning on their camera. I want to just take a general screenshot. Yes.
Is it fine? Can you see
Ashita, are
me?
you there? Yeah, I can see you. Please just turn on your camera. I would love to just take a screenshot real quick. Yeah, thank you so much. Thank you so much for those. Wow, so lovely seeing a lot of faces. Yeah.
Can you see
Yeah,
my?
please.
Is it fine?
1 minute. No, it's not on. Wow, yeah. It's quite okay. Dr. Adekunle, if you can also take a screenshot, it will be so lovely. I can't see you, sir.
Yeah,
I can't see you, sir. All right.
The camera is on.
Is it fine? Can you see me?
I can't still. Wait, I can't see Dr. Abdulaziz too. All right, but I will still take a screenshot nonetheless. I'll take a screenshot nonetheless. I will take a screenshot nonetheless. Dr. Adekunle, you two please take a screenshot from where you are. Thank you so much, everybody. I've taken a screenshot. Thank you so much, everybody. I've taken a screenshot. I really appreciate your presence.
Okay.
Now we can see you. Okay. Let's take the last screenshot. I'm sorry. Let's take the last screenshot again. Some person's camera just connected. Dr. Shola Ojo, please. Some person's camera just came on now. Yeah. Let's take that screenshot again. 3, go. All right. Thank you so much, everybody. It's so lovely to have everyone here. I'd love to call on the next section to take over our Asian team. Are you there? Or Asian team, are you there?
Yes, I am here. So thank you so much, everyone. Let me quickly share my screen. All right, so thank you so much, Dr. Faluke, Dr. Anjurin, Mr. Ashraf, Mr. Adloye, and Mr. Ayafe for handling the Omics Logic Africa very well. And it was pretty insightful and informative session to learn about the various challenges and opportunities that the session had to explain about. So before we quickly wrap up today's day 1 of Omics Research Symposium, let me quickly give you an overview about tomorrow's session schedule. So in day 2 of Omics Research Symposium 2023, you will hear from other experts from Asia, Africa, and USA. So first of all, we will be joined by Dr. Jagdish Gandla, who will be discussing about trends in healthcare and biotechnology. Dr. Prasanna Espoti will help you gain valuable insights and knowledge about how bioinformatics is being used to shape the future of academic and industrial research. Ms. Anuja Mishra will also introduce the audience to the latest guidelines and research that are shaping the future of medical genetics and genomics. You will also hear from Mr. Emmanuel, who will share insights on latest training and capacity building opportunities. Ms. Elizaveta will also take the participants to the world of single-cell datasets and discover how they are transforming our understanding of prostate health and disease. Dr. Laura Harris will introduce participants to integrating multi-omics data and conducting meta-analysis. You will also hear about the prize distribution ceremony from today's poster presentation competition. From Ms. Sonali Kare, you will learn about bioinformatics education and groundbreaking work that is done at Omics Logic America. You will also learn about the present status of data science and bioinformatics in Louisiana and IDA states from Dr. Gus Katsoulis. Dr. Jamie Souza will also help you delve into the world of next-generation sequencing and its application in the study of animal and infections. So we look forward you all to the rest of this symposium and exciting discussions and insight that are yet to come. So thank you everyone for participation and contribution, and we hope that the symposium will lead to further collaborations and advancements in the field of bioinformatics and data science. So see you all for day 2 of symposium that is scheduled for 5 p.m. IST, 6:30 AM CST, or 12:30 p.m. GMT. Thank you everyone. Have a great day.
Thank you so much.
All right. Thank you, Shiraguri. Thank you, everyone. See you
thank you
tomorrow.
all.
Thank you, everyone.
Yes, thank you.
Thank you. Enjoy your day.
Thank you. See you tomorrow.
Thank
Yes,
you.
thank you. See you
10:30.
tomorrow.
Ayanfe, take care of yourself.
Yes.
Yeah, thank you so much, Doctor.
Thank you
Yeah,
very
thank you.
much. Bye-bye.
Professor
Thank
Kulu,
you.
Yeah, thank
bye-bye.
you so much.
Thank you.
Prof, well done, no, Professor.
Thank you,
Thank
Prof.
you, sir.
Yeah, okay.
We'll
Thank
connect
you.
and take further
Thank you.
discussion.
In University of Ilorin.
Thank you.
Okay.